Thanks for coming, everybody who showed up. So before we start with the class itself, I'd like to show you how we broke down the topics to fit into our schedule. So as we mentioned, every class is 4 hours and 2 classes a week for 6 months. If you do the math, it'll turn up to be around 200 hours. So we split the topics in a manner in which we can fit everything in 200 hours. So I'll show you the split up right now for the first few classes and you get a better idea. Won't take much of your time. Can you see this?
Yeah.
Yeah. So every class, we'll be discussing one system and one extra topic. So the most important topic of the most important subject for this is obviously pathology and pharmacology, right? And that is that we are going to discuss that system wise. We're going to, like, let's say we take cardio, we do its pathology, we do its pharmacology, also touch on its anatomy and histology and all the other basics. But with one system, we'll also be doing another topic that is not related to the system, like micro. It doesn't fall into any system in specific. It's a topic of its own. Biochem, micro, ethics, and immunology are separate topics. So in every class, we'll divide our time in such a manner that we discuss one system and one topic other than the stuff pertaining to that system. So basics of Path and Farm will be our first topic. We had mentioned in the email that for today we'll be doing basics of Path. We've given it 8 hours, and so you can basically. So as a total, we have 200 hours. We kept a small buffer, so this won't completely add up to 200 maybe, but this is what we plan on doing. We plan on spending 8 hours on basics of Path and Farm respectively, and then moving on to cardio, which has a lot of weightage when it comes to the number of questions, and it is very concept heavy. So we thought it needed more time, and we gave it more. Endo and psych relatively have relatively lesser portions, and they can be completed in a short amount of time. So we took some time off both these subjects to give it to cardio, and everything else will get 16 hours. Every other, like pulmonology, nephrology, and all of that will get 16 hours. Micro, we'll teach you the basics today, and we'll take 2 hours for that. Immunology, again, we'll take 6 hours. Epibio and biochem will take 12 and 10 hours respectively. I already mentioned for the first class, we're doing basics of pathology and micro. So today is just one class, but usually in a week we'll have 2 classes. That's why this is adding up to 8 hours. So for today, it'll be 3 hours of path and 1 hour of micro. But if it was in a weekly structure, it would be 6 hours of path and 2 hours of micro. Same goes for the third and the fourth class. It's totally adds up to 8 hours in a week. So in that week, we'll be doing 6 hours of farm and 2 hours of immuno. After that, we'll be starting with the most important system, which is cardio, and we'll also be doing immuno together with that. Same goes to the next class because cardio has 18 hours, right? So we had to give it 3 weeks. And also immuno has 4 hours. So we had to give it 2 weeks. And finally, we'll be starting with biochem. And we'll start off with 2 weeks and 2 hours, and we'll see where it goes from there. This is the structure for the first 10 classes, and we'll build on the rest later as it comes. Yeah, that's all from my end. Now, Abhi will be starting with the portion.
Before we start with the lecture, do you guys have any questions regarding the class structure or like what we will be covering in the topics?
I have no questions.
Okay. So yeah, I guess we'll just begin with the presentation. So today, the first lecture will be on cellular injury, which is a very important part of basics of pathology, and we'll be covering some concepts first of cellular injury. Then we'll move on to micro, like Ashwin said, 1 hour of micro almost, and then we'll have we collected some questions from UWorld and Ambos, so that we'll discuss the questions with all the right and wrong answers at the end of the class. So whatever we're going to study in cellular injury, you'll see USMLE based questions that you'll be asked on the same topics. So let's move on. Oh, and also one more thing, it'll be better if you guys can take some notes because in the presentation we have included high yield information like examples of pathology that you might not see in every resource. Like we combined First Aid, Pathoma, Amboss Library, and a few other sources. So this might be helpful if you take down the examples at least. Okay, so we'll begin now. So first we'll start with growth adaptations. So in a normal cell, what we know for a fact is it's always in homeostasis with the environment. Homeostasis means it's in an equilibrium with the environment, whether it be with pH or the concentrations of potassium, sodium. All these ions are in an equilibrium. And the thing is, the environment keeps changing. That's a given fact. So our cells have to be able to adapt to the environment, and there are different ways that the cell can adapt. So the environmental stimulus that triggers these adaptations is called a stressor. So this environmental stressor can cause different changes, and these different changes that you can see in the cell, there are many more, but the ones that are high yield and like a lot more tested are hypertrophy, hyperplasia, atrophy, and metaplasia. We'll go through each one of them and like how they work and what it means to the cell and examples of each of those and when they are bad. But the main thing you have to understand is it's better than memorizing these words. If you know the meaning of the Latin roots of these terms, it might help you remember them better. So I mentioned that stresses, and then we'll have adaptations by the cells to deal with the stresses. But sometimes, if the stress is too much, it's something that the cell cannot respond to. If it exceeds a certain threshold, then there will be cell injury. Because example of this would be like if you drink. Okay, this would normally happen, but there are questions regarding these. So if you drink like what do you call bleach, if a person drinks bleach, it is a very caustic substance, and our cells once they are exposed to bleach, they will start disintegrating. They don't have time to adapt to such an acute stress of a stimulus. But at the same time, we have things like people able to live in high altitude conditions, because over time they because it's a gradual uh, development, because people over the millions of years they have developed the ability to live in high altitude conditions, so it has triggered changes in their hemoglobin and oxygen carrying capacity. These are like adaptive responses that the body goes through if it's given enough time, and if the stimulus does not exceed a certain limit. If those things happen, it will result in cellular injury. And obviously a normal cell, if there's a very toxic stimulus, it can directly result in cell injury. Or even if these adaptations happen, but if these adaptations are not enough to even deal with the stress, so over time, yeah, we'll discuss the examples with each one specifically. So first one we have is hypertrophy. So the main thing that you have to know about this is the word. The meaning of the whole thing will be given in the word. So you have trophy. Trophy is from a Latin term for trophic, which means growth, and hyper you all know, which means more. So this basically means more growth. But how can a cell increase in I mean, how can a cell grow? It has to increase in size. For it to increase in size, it needs to produce more protein and organelles. Because if the cell just increases in size, it cannot sustain all the active transport mechanisms or the energy requirements of the cell. So it would need to produce more mitochondria. And if there's more cell membrane, then it needs to produce extracellular membrane proteins, intracellular membrane proteins, and a lot of stuff needs to happen for a cell to increase in size. So the exact mechanism by which hypertrophy occurs in a cell is, first there'll be gene activation. Maybe there'll be some stimulus from the environment. Maybe it might be an increased pressure signal, or it can be any signal that triggers a gene to activate. Not just one gene, many genes that are involved in making cellular proteins. and this will be transcribed to mRNA, and then you'll be produce more proteins. The details of how transcription and all that, don't worry about them, because we'll definitely cover them in molecular biology in more detail. But just know for hypertrophy that you'll have increased protein synthesis because of gene activation, and then because proteins are produced, also you need to produce more organelles to deal with the proteins, like, and mitochondria for ATP. So you'll have increased number of mitochondria, increased number of endoplasmic reticulums, and those organelles as well. So this is the basic mechanism behind hypertrophy, and it's an important thing to know, as you'll see in the questions that might come later on. And hypertrophy can be of two types. It can be physiologic or pathologic. So physiologic means that it's something that occurs normally or in our body, and it's a normal response, or it's something that is normal. In people who exercise, you can see that their muscles start to develop and they start to grow. So this is an example of hypertrophy, because smooth I mean skeletal muscles do not divide, so they only undergo hypertrophy. That's another thing you have to keep in mind, is that there are some cells that cannot divide, and these examples is something you have to know. These are high yield information. So there is something called permanent tissue, cardiac myocytes, skeletal muscles, and nerve cells. These three cells do not divide. So if there's an injury to them, there will be a decrease in the number of cells, and if they have to develop more, it would only be hypertrophy. You cannot have increase in the number of their cells. And it's also important to note that most cells are not like this. So it's just cardiac myocytes, skeletal muscles, and nerve cells. So every other cell can also increase in cell number. So most of the time, hypertrophy and increasing in cell number, which we'll look at next, hyperplasia, occur together. So keep that in mind. And an example, this is an example of hypertrophy in a clinical context. So if a patient has hypertension is high blood pressure. When there's increased blood pressure, your body has to respond to that. Obviously, the blood pressure increase has to be, like we said before, it has to be in a small increment. It cannot be like a sudden, it goes from like 120 systolic to it goes to 240 systolic within a matter of 2 hours. That means you'll definitely get a stroke. Your body does not have time to adapt. But suppose over a period of 5 years, this patient has been going from 120 to 180 to 200 of blood pressure, then your body does have time to adapt. Your heart has time to go undergo hypertrophy, meaning the it'll produce more mitochondria. It'll pump against a much higher afterload, which is the increased blood pressure, and your body will adapt to that state. So this pathologic change in the heart that occurs due to hypertension, because it has to pump against a large amount of blood pressure, is called hypertrophic cardiomyopathy. And there are different causes for it, but one of the causes is hypertension. Do you guys have any questions regarding this topic? If you have any questions, you can just interrupt me, because I can't see the Google Meet screen right now. So these are the different changes in a diagram form.
I have
So
a question.
yeah, sorry.
You had said that hypertrophy is one of the cause of cardiomyopathy, right?
Mhm.
Isn't that isn't the cause will be different, and our body is responding to that cause by doing hypertrophy, right?
Yes,
Or
that is
the
correct.
main cause?
That's correct. So as you can see here, yeah, I might have forgotten to mention that, is that hypertrophy, it's an adaptation to deal with the hypertension. But then once you have the hypertension for a long time, what happens is your heart cannot deal with that for a long time. It meaning it's consuming a lot more energy. It's much thicker than it used to be, so its blood supply is also less. So eventually the hypertension will be too much for the heart to deal with, and it will lead to failure. So that's what chronic hypertension results in. First, hypertrophy of the cardiac myocytes, and then when it fails, or uh, when there's a disease, meaning the patient has symptoms because of the heart not being able to cope up with the stress, which is hypertension, then you'll result in the clinical disease called hypertrophic cardiomyopathy. So in here it's mentioned, as you can see, adaptive responses when they are exceeded. So the hypertrophy is not enough to deal with the increased blood pressure basically. Does that answer your question?
Yes, yes, it does.
Okay. So this is a diagrammatic, yeah, representation of the cellular changes, the adaptations that we saw. As you can see, a normal cell, if I mean a normal tissue, if it has 4 cells, what would happen is in hypertrophy, they would increase in size. So as we mentioned, increasing in size happens because of increased protein synthesis and organelle production. These two are really important to keep in mind because they differ between each types. Next, we have hyperplasia, which we'll be discussing next, which is basically increase in the number of cells. The size doesn't change, but the increase in number of cells. Atrophy, the cells shrink. We'll go through all of them in detail. I'll just cover them slightly. Metaplasia is that they change in the cell type. So if these were suppose cuboidal epithelium, they change into like squamous epithelium. And dysplasia is when it loses its original shape or function some due to some mutation. So we'll move on to hyperplasia. Hyperplasia again, if we look at the word, it might help in remembering. So plasia comes from plastic, which is like molding, and hyper, so which is more molding or forming. So this means there's an increase in production of new cells. So this can obviously happen only in some parts of the body. So as we saw, there's cardiac myocytes, nerve cells, and skeletal muscles which cannot undergo hyperplasia because they don't have the required stem cells. Usually, epithelium or some mucous membranes, these have a high number of stem cells and they can undergo hyperplasia. Even examples we'll look at in a bit. Um, so an important thing to keep in mind is that in hyperplasia there are stem cells, and from stem cells they are given a signal to increase in the number of cells. So they undergo cellular replication. So what we know from like basics of molecular biology, I'm sure you guys studied before, is which we will also cover. But just want to mention that if there is an increase in replication, then the enzymes that are involved in replication always have like a fidelity rate. A fidelity rate means the amount of mistakes they make per maybe like 1 million nucleotides or something. And our whole DNA is about, it's pretty big, around billions of nucleotides. So what happens is if there is a fidelity rate, meaning amount of mistakes they make per number of nucleotides, then there will be mutations that happen. But our body is pretty good at controlling these mutations. We have uh, mechanisms to repair the DNA damage that occurs during normal replication, and usually mutations are not seen. But if there's increased number of replication from stem cells, there's a high chance of making more mistakes, and this can eventually, if it lasts long enough, meaning if the stressor that is causing the hyperplasia is it lasts long enough, it can lead to dysplasia. Dysplasia, which I'll explain next, but is something that happens when the cells have too many mutations to have their previous function. They cannot perform the previous function that they were able to do properly. And eventually, dysplasia can accumulate even more mutations, maybe some tumor suppressor gene, and then eventually lead to uncontrolled proliferation and cancer. Again, we'll look at those mechanisms in a bit, but I'm sure you guys are able to understand this one. Yeah. Okay. Um, So hyperplasia, we have two types again, physiologic and pathologic. So physiologic hyperplasia you'll see in a lot of different organ systems. One example that we have here is during menstruation. So what happens during the menstrual cycle in females is in the uterus there's an endometrial lining. So endometrial cells, what they do is over the course of the menstrual cycle they are stimulated by estrogen, and once they're stimulated by estrogen, because endometrium has a basement membrane filled with stem cells, so what happens is once the estrogen goes and activates these stem cells, it gives a signal for them to divide more. So what happens is they divide over the course of the uh, menstrual cycle, and during the end of the menstrual cycle they undergo atrophy, meaning they shed, which we'll look at. But the main thing is that hyperplasia happens during the menstrual cycle because of estrogen. Estrogen is another stimulus for hyperplasia. So this is physiologic, meaning that during the menstrual cycle there are normal variations in the levels of estrogen, and this causes the endometrium to proliferate. This is called this is in normal non-pathologic cases. Then there's another type in bone marrow. Bone marrow is filled with you guys know of like the precursors of platelets, precursors of red blood cells, and the precursors of uh, white blood cells, and also hematopoietic stem cells. So what can happen is, suppose your red blood cells are getting destroyed for some reason in some kind of hemolysis. It can be autoimmune hemolysis for any cause, maybe because of sickle cell anemia, malaria, some cause of hemolysis. What happens is the bone marrow has to respond. They have to increase the amount of RBCs in the circulation, because it's not enough for the body to live on. So what happens is the bone marrow undergoes hyperplasia. So the amount of the stem cells will start to divide, the hematopoietic stem cells, and they will give rise to increased number of white blood cells, red blood cells, and also platelets. So pathologic cases of hyperplasia, we can see that in the endometrium again. So I mentioned that estrogen is the stimulus for endometrium to grow. So this estrogen has a normal variation. It's not always high or it's not always low. Throughout the menstrual cycle, because of different hormonal stimulations, they go up and down. So in cases where estrogen levels are consistently high, so this can be seen in different groups of people. Maybe if someone has an estrogen secreting tumor, so there will always be a consistently high level of estrogen. This will cause the endometrium to always have the signal of growth, and this chronic stressor of estrogen can lead to dysplasia because of mutation accumulation, and then eventually cancer, endometrial cancer. So this is one way that endometrial hyperplasia can lead to endometrial cancer, because of chronic high stimulation from another estrogen secreting tumor. So an estrogen secreting tumor, by the way, is a risk factor for endometrial cancer, just because estrogens are consistently elevated. Another good example is seen in obese obese women, is because adipose tissue has an enzyme called aromatase, which can convert androgens, meaning testosterone and other androgens, into estrogen. Yeah, they can convert it to estrogen, adipose tissue. So because of the excess adipose tissue, there will be excess peripheral conversion, meaning periphery in the adipose tissue. There will be conversion of uh, these androgens into estrogen, and these estrogen will then go and stimulate the endometrium, and that's why obesity is a risk factor for endometrial cancer. That's also a high yield point to know. So these small examples that we mention in between, it might be a good idea to write it down, because these are what the questions will be based on. The questions won't just ask you like what is hyperplasia. They'll give you a case of like a 50-year-old woman who's obese, smokes, and African American. She will come, and then you will do an endometrial biopsy, and there'll be hyperplasia, and then they will ask you probably what is the risk factor, what is the major risk factor for this patient to develop endometrial hyperplasia. So the answer would be obesity, because obesity, again, sometimes they might not just ask you obesity, what is the mechanism by which she develops endometrial hyperplasia. So then you should know that obesity, in obese woman, there's adipose tissue, and then adipose tissue has this enzyme that converts androgen into estrogen, and because of that, estrogen stimulates there's continual stimulation of the endometrium, leading to endometrial hyperplasia. Just how you should be thinking about questions as well. And the same thing can happen in men in the prostate. So BPH is benign prostatic hyperplasia. Again, don't worry that much about the mechanisms and details. If you're not understanding, we'll definitely cover them during when we are discussing the disease itself. Just a small idea. So in benign prostatic hyperplasia, what happens is prostate gland, which is prostate gland, its main function is to secrete prostatic fluid. So the prostate gland is stimulated by androgens, so like testosterone and dihydrotestosterone. What happens is, over time, especially as men get older, there is increased stimulation of androgen, and this can result in the prostate to undergo hyperplasia. So that's why BPH, if you look at the statistics, men over the age of 80, there's a 90% chance that they will have benign prostatic hyperplasia, because all over 80 years they have had stimulation of their prostate gland, so it is bound to undergo some hyperplastic changes over time. So this is seen in BPH. So it's also an important fact. This was mentioned in Pathoma as well. And whatever small detail that you find mentioned in Pathoma and first aid is always important to keep in mind. And that means that hyperplasia, what's mentioned was hyperplasia leads to cancer in most cases, but except in BPH. So endometrial hyperplasia is a risk factor for you to develop endometrial cancer, but benign prostatic hyperplasia is not. This is like one exception. So it's always, when there is an exception, it's always important to keep it in mind, because they will test that, because they know that people think that okay, hyperplasia means more mutations, then eventually dysplasia, cancer. But there are some exceptions, and this is one of them, one of the most important exception, BPH. Do you guys have any questions till now?
No.
All right. Next, we have atrophy. So, atrophy, again, the same thing we'll do with the word trophy. Trophic comes from the Latin name for growth, and A is like a negation. So, there's decreased growth, or yeah, reduced growth. But one thing you have to differentiate from hypertrophy and atrophy is that atrophy happens from both sides, meaning decrease in cell size and number. But hypertrophy is only increase in cell size. This differentiation, just keep in mind, because sometimes later on you can get confused. Atrophy, there's decrease in cell size and or cell number. So causes of atrophy, there are many causes of atrophy, and even you guys can see a real life example, disuse atrophy. So if you see the difference between the muscles in your right hand and your left hand, you know that they are different, because it depends on which is your dominant hand. The muscles in your dominant hand will be more developed than in your non-dominant hand, because you use the muscles more. So not only will the muscles in your dominant hand undergo hypertrophy, they can also the muscles in your non-dominant hand can undergo atrophy as well. So meaning you use it less, so there's less stimulation, and eventually the proteins will get degraded, and the cell will lose their size and number depending on which type of cell. So if there's a loss of blood supply, you can imagine, if the cells are not getting their nutrients, obviously they cannot produce enough ATP to maintain the enzymes and all the processes that are happening. So they will start degrading all those uh, useless ones first, and eventually it can lead to a decrease in cell size or number. Denervation is also an interesting one. So we have muscles, yes, and they need the blood supply, so muscles will function. But the main thing to direct the muscles is the neural impulses. So a brain or the spinal cord has to give the signal to the muscles to tell them that you have to contract at this time, or this is what you have to do. So if there's some damage to the nerve, so maybe like during a car accident, or if there's some inflammation of the nerve in like multiple sclerosis, what happens is the muscles lose the signal that they receive, and this can once they don't receive the signal, that means they are not stimulated to contract, and if muscles don't contract, they are not being used, and eventually it goes through the same process. The proteins that are not needed will get degraded, and eventually it'll undergo atrophy. And again, lack of hormonal stimulation. So as we saw in a small example previously, it was estrogen stimulation on the endometrium. So if there is no estrogen, suppose in like Turner syndrome or something, in which they don't have ovaries but they still produce estrogen, that's a bad example. Maybe some case in which the patient has decreased amounts of estrogen, then that can result in lack of endometrial proliferation, things like that. Poor nutrition is the say, it's kind of related to blood supply. So if a malnourished person, you can see there'll be like generalized atrophy of their muscles, and yeah, of their muscles. You can see that as an example. So the mechanism, these two mechanisms sorry, these two mechanisms, they're really important to keep in mind, because whenever there's a proven mechanism that is widely recognized, it is the USMLE step one is bound to test it, because these are some of the sure questions that they can answer without a doubt. Because there are other mechanisms that, like, sometimes it might be true, it's not true, we don't know. Most of the things are like that in medicine, but when there is proven mechanisms, the USMLE step one would love to go after mechanisms. So it's important for you guys to keep in mind. So the two mechanisms that I'm going to talk about, they're really important, because one won the Nobel Prize in 2004 and in 2016. I searched that up. So these mechanisms definitely will come one way or another in your exam. So first mechanism is how the cell decreases in size. So a cell can decrease in size. What we talked about, just the basic logic is that if a cell is too big and if it cannot sustain itself, either because of disuse, loss of blood supply, something, so it has to reduce its size, because it's not producing enough ATP. It's not able to sustain the cellular processes, like maintain the sodium potassium pump, maintain the normal concentration gradient. So the cell has to decrease in size to allow it to survive better. So this can happen through degradation of the unwanted proteins. So so the main mechanism is it degrades the unwanted proteins. So just focus on the top part. So this is the mechanism by which cells degrade proteins. So it's called the ubiquitin proteasome degradation pathway. It sounds complicated, but it's very simple. It's just the big names for things that they have given. That's it. So basically, you have an unwanted protein. So you can ask, how would a cell know what's an unwanted protein? Basically, it all depends on signals. So if there is a signal for the cell to degrade a certain type of protein, which can come from extracellular, intracellular. So what I mean by signal is, remember when I said estrogen is a signal to stimulate endometrial proliferation. Suppose there's a loss of estrogen, then it's a signal for that proteins in the endometrial cells to get degraded. So what they would undergo is, how a cell goes about doing this is, there are these three proteins called ubiquitin E1, E2, and E3. You don't need to know the details about this. Just know that there's this. Uh, there are these proteins that complex together. They form something called a ubiquitin ligase, and what they do is they take ubiquitin, which is a small protein, a small peptide, uh, of like, yeah, a small peptide. It's like a signaling marker. They add it to the protein. So the main function of ubiquitin ligase is to ligate the ubiquitin to the protein. Okay, it just doesn't do it once, because there's a difference between having one ubiquitin on the protein and multiple. So it's like a different signal for the cell. If a protein has one ubiquitin, it is bound for something else, versus if it's polyubiquitinated. Polyubiquitinated means it just has multiple tags on it, and once a proteasome sees this signal, what happens is it degrades it. So this signal must be present for the proteasome to degrade this protein into peptides and amino acids. So you guys understand how this mechanism works, yeah? Any questions on this?
No.
Um, all right, so that is how it decreases in cell size. So ubiquitin proteasome degradation. So that is the mechanism. And what protein it degrades the intermediate filament of cytoskeletons. So cytoskeleton, so cyto means cell and skeleton is like the backbone. So the backbone of the cell has different types of filaments, which we will cover later on, but just know it has like a mesh of stuff that is holding the cell together from the inside and the outside. So what happens is, if you want to decrease the cell size, you need to reduce the frame size. Yes, so these intermediate filaments get tagged by this ubiquitin pathway, and then it goes to the proteasome and it gets degraded. So the frame gets smaller. The cell can easily get smaller after that. And another mechanism is autophagy. Autophagy, Ashwin, are you going to be discussing autophagy later on?
Yeah, I'll touch upon it. Yeah.
Okay. So autophagy, Ashwin will be discussing that in a bit. So I'll just mention that autophagy means it's self-eating. So to degrade the cellular components, it basically just eats its own component. Eats meaning digests using like acidic enzymes and acidic environment and enzymes, which you can see in lysosomes. That is one way in a cell can decrease in size. And cell number, you go through apoptosis. Apoptosis, Ashwin will be covering in very good detail in a bit, but apoptosis, I did not look up the Latin meaning for it, but I am pretty sure it would probably mean something along the lines that it is programmed cell death. So
Yes, suicide.
yeah, so programmed cell death would be so the cell itself decides that it doesn't want to live, probably like suicide would be a good example. So it decides it doesn't want to live, and then it initiates the process which will eventually leads to its own demise. Ashwin will touch it in detail. What happens, there are other things that happen in atrophy. There's something called involution. Involution is like when the organs which suppose they are enlarged, or they just involute, meaning like they undergo degradation via some kind of atrophic process, and it loses its original size, and sometimes it might even be that they don't function. So physiologic aging, which you can see in senile atrophy. I have some good examples of atrophy for you guys, but senile atrophy meaning like over time because of old age. Old age means one important thing you guys have to keep in mind about old age means the same thing that we talked about in hyperplasia. Hyperplasia is increased number of replications. Old age is the number of years that they have lived increases, so the amount of replication that happens also has been increased. So that means now chances of mutations are also higher. So this can lead to some kind of atrophy because of mutations and other causes. That is senile atrophy. So pathologic causes of atrophy would be like if someone has malnourishment, they can be generalized, they can be atrophied everywhere. And obviously if you don't use a particular limb or something, they can be atrophied. If there is pressure, so people with if you apply pressure on one area for a long period of time, like I'm talking like 6 months, 1 year, that area might undergo atrophy because of the pressure on it. This can be seen in like ICU patients who have been on like bed rest for years, or like weeks and no months, sorry. This can you can see behind like in their toes and all the places where they are contacting the bed, you can see some form of atrophy in the muscles um, in those patients. And also nerve damage, you I explained before, like if they don't have the nerve stimulus, they cannot contract. And chronic inflammation we'll touch up, that will be our next topic, so I don't want to go into that detail on it. But chronic inflammation can also lead to atrophy because of scar tissue. Yeah, so here we see two hands. Can anyone uh, tell me what you guys see the difference between these two hands are? Mm, okay, to help you guys out, you guys can focus on the right next to the thumb, like where the arrows are pointing. Do you guys, can you see the difference in the muscles in these area? So this muscle is the it's a group of muscles in the thumb. It's called the thenar eminence. This it's a group of muscles. There are three or two muscles in that control the movement of the thumb. And as you can see in this patient, there is a decrease in size, meaning atrophy of that muscle. So can anyone tell me what do you think might be the cause that only one of her or his hand has atrophied compared to the other? Any guesses regarding the cause?
Is it pressure?
It could be pressure. Pressure is a good one. The disuse also could be a good one because it's local, but it's unlikely to be senile, because senile atrophy you would probably see it in both hands. So it can be either disuse, pressure, but in this case, this is because of denervation. This is a syndrome called carpal tunnel syndrome, in which the motor neuron which is supplying to the muscle, your thumb muscle, which is coming through in your wrist, is getting squeezed. So the muscle loses its innervation, and eventually it leads to atrophy. So this is a good clinical example of how atrophy happens in carpal tunnel syndrome. Next we have Next, we have this slide of histology. This is cardiac myocytes, and as you can see, these are normal cardiac myocytes with some kind of brown color pigment in them. So one thing that this is a important thing to keep in mind is that over time there will be over time, as in like as people age, there's a lot of wear and tear, and especially in places like cardiac myocytes which don't regenerate, there will be a lot of damage occurring. So you know that cells are made of a phospholipid bilayer. So phospholipids, what happens is over time, because of oxidative damage, they undergo peroxidation. And once they undergo peroxidation meaning like they are attacked by oxygen free radicals, which again we'll be discussing in a bit. But these chemical reactions that happen cause the some parts of the cell membrane and some proteins to degenerate, and these fatty acids, they will clump together and form something called lipofuscin. So this is an important term you guys have to remember. It's called lipofuscin, and that's the name given to these brown spots that you can see in these cardiac myocytes. You're not only in cardiac myocytes, you can probably see them in everywhere in your body, and usually in old people. This is not pathologic. There's no, like, it doesn't cause any symptoms, and mostly it's found in autopsies. When they want to look for another cause, they can find this and they'll say, okay, this person has is probably like really old. That's the point regarding this, because sometimes they'll give you a histology slide like this and ask you what is the cause of the brown stuff. So you might think it might be pathology because it's specifically highlighted, but it's not. It's a normal thing seen in aging people. And the pigment, just keep in mind, it's lipofuscin, and it's due to peroxidation, because it's not just enough to know that this is lipofuscin. You have to know the mechanism by it happens, because it's oxygen free radical damage to phospholipid bilayers and other stuff inside the cell that causes peroxidative reactions. Next we have metaplasia. Wait, so far do you guys have any questions? No. All right. I'll move on. Metaplasia. So this is an interesting one. This has the most number of examples and the most number of questions also most probably will be coming from metaplasia. So metaplasia is basically a response uh, to stress, again, which is what all the adaptations are, but this response is a bit different. First we saw increase in size, cell number, then we saw decrease in cell size and number. Now this is a whole new change in cell type. So again, if you want to change the cell type, that means they have to be able to have stem cells. So definitely you won't see metaplasia in the heart, in the skeletal muscles, and in the nerve cells, because they don't have stem cells. So in places where you do have stem cells, this chronic stress can cause them to get reprogrammed and adapt, because stem cells are pluripotent. Pluripotent meaning that they can evolve into different other cell types. So depending on the signal. So what happens is this chronic stress acts as the signal to reprogram the stem cell, and it will change the type of cell it is. Suppose it was squamous epithelium, it can become columnar epithelium or intestinal epithelium, things like that, depending on which environment it has to deal with. Uh, so that, and this change is always to tolerate the adverse environment. So again, this is initially it will always be an adaptive response, then eventually once the stressor is too much or there's some mistake that happens in between, this will lead to like cancer or dysplasia. uh, Metaplasia, it can completely regress. So if this for so the stressor causes, suppose, a cell to change from cuboidal epithelium to stratified squamous epithelium, then once you take away the stressor, then eventually the cell will revert back. There is theoretically it's possible for them to revert back to normal. Not just theoretically, yeah, it can regress. And or if it doesn't, or if the stressor doesn't go away, if there's continuous stress on that tissue, then it can lead to a persistent insult and then eventual malignant transformation. So malignant transformation, if there's enough wrong mutations that are accumulated, dysplasia, and it can be considered precancerous. So there's one exception to this. So again, metaplasia can, so this is how it would go. So first, there's chronic stress. An example I'll give you in a bit, but there's a chronic stress, stem cell get reprogrammed once the stem, and then it changes to a different cell type, and then that cell type keeps still having more and more insults. So the signal keeps coming to the stem cells and they keep dividing. Eventually there's a mistake and it can go to dysplasia and then cancer. So most metaplasia is considered pre-malignant, you can consider them, but precancerous, except apocrine metaplasia of the breast. So since this is an exception, you have to know this. Apocrine metaplasia of the breast, it's probably just a bunch of words for you guys now, but we will discuss breast pathologies later on. But what you have to know is there are apocrine glands in the breast. So these glands can undergo changes based on the hormonal stimulation, and because of this stimulation it can undergo metaplasia, and it is not cancerous. That's all you have to know. We'll touch up when we come to breast pathology. I'll remind you guys about this again, definitely. So one of the most important clinical manifestations of metaplasia is called Barrett's esophagus. I'm sure you guys have heard of Barrett's esophagus. So what happens in Barrett's esophagus is there's chronic gastroesophageal reflux. So as acid in the stomach gets refluxed up into the esophagus, and the acid irritates the lining of the esophagus. Usually, the esophagus has the lining is called stratified squamous epithelium. Stratified squamous means there are layers of just squamous epithelium, and what happens is it's not meant to deal with acid, this kind of epithelium. Our intestines and the epithelia in the stomach, on the other hand, are meant to deal with acid. They can deal with pH of even 1. So what our body does is, oh, okay, I'm seeing acid right now, so the acid acts as a stimulus to convert the stratified squamous into something it can deal with the acid, which is intestinal epithelium. And that is the transformation, and that transformation is called Barrett's esophagus. The problem with Barrett's esophagus is that if that acidity continues almost for 5 years or something, and this metaplasia has happened, eventually there can be a mutation that causes its transformation to a dysplastic epithelium, meaning there's mutations that cause increased cell division and eventually cancer. So that's why chronic acid reflux, chronic I mean like almost 5 years of continuous acidity every night over the month, over the year, would lead to esophageal adenocarcinoma because of this Barrett's pre-malignant condition. Another good example is how smoking can cause squamous cell carcinoma. How it happens is our respiratory epithelium, meaning the epithelium in the bronchi and bronchioles, they are lined by pseudostratified ciliated columna. So all that big word, what it means is that it's a cell that is in the shape of a column and it has cilia on top of it. So these cilia help in like moving the mucus out of the bronchioles and help trap like bacteria and other pathogens. What happens is once you start smoking, the smoke is an irritant to the cells lining the respiratory epithelium, and then this causes it to create more defense. And as you can see, stratified squamous is what you see in most outer parts of our body because it's a good defense, because there's like thick layers of thick layers of cells with like dead cells on the top, so it's a good defense. So that's why once people start smoking, the respiratory epithelium thinks that, okay, I need something to defend against this smoke irritating me, so I'll just switch to stratified squamous epithelium. Then once stratified squamous epithelium happens, then the same principle. Then the smoke keeps on pushing the cell, causing mutations because of replication. Eventually, it leads to cancer, squamous cell cancer. Same thing, there's a condition in the bladder that can also be seen. Another important condition that you see metaplasia is in vitamin A deficiency. So I have pictures of these. So yeah, let me just go through this histology slide. I'll also introduce you guys to how to read a histology in each whenever I show you a histology slide. So what you can see here is the stain they done on these cells are called hematoxylin and eosin. It's the common stain that is mostly done, H&E stain, and it usually colors them pink and purple along those lines. So here you can see it all looks very chaotic, but if you can see, there is a differentiation between the lining of cells here and over here. So this area with like multiple layers is the stratified squamous. So there are multiple layers of cell over, and then as you go over them, you can see these are more dead cells because they don't have nucleus on top. But as you can see in this area, it's more single layer of cells lining the outer part. So uh, these are the intestinal cells, columnar cells that we saw. So this is a picture histological slide of the gastroesophageal junction and how it has changed because of Barrett's esophagus. All right, so this is the normal epithelium in a esophagus that you'll see, and this is the change that has happened in this patient because of acid. So your cell types switch from this to this because one thing to know is that the stem cells for this squamous epithelium are actually located at the first layer of cells at the basement membrane, meaning like this thing that's separating the connective tissue in this area is where the stem cells reside. It's important to know that as well. That's that. This is another example. As you can see, this is but this is the opposite. This is change from the normal cuboidal epithelium into stratified squamous. So the pathologic change is the stratified squamous. This you can probably see in the lungs, in the respiratory epithelium. Sorry. Okay, so this is the metaplastic. So it changes from this cell to this cell type. This you can see. This is something called a Bitot spot, BITOT, and this this spot you can see it in vitamin A deficiency. Vitamin A deficiency. So I won't go into too much detail, but vitamin A deficiency, what it happens is it causes vitamin A is necessary for you to maintain normal keratin and your epithelial lining. So what happens is once you have vitamin A deficiency, you cannot maintain the normal epithelium. So it undergoes like keratomalacia, and you can see excess keratin on your eye because of vitamin A deficiency. So they might ask you a question like, what is the mechanism behind uh, Bitot spots in vitamin A deficiency? Or they might just show you this picture and ask you, what is the mechanism behind the formation of this white spot? So it would be metaplasia due to vitamin A deficiency. This is another thing. It's called myositis ossificans. So just the name you see, myositis. Myo is muscle, itis is inflammation. So inflammation of the muscle. Ossificans means ossification, bone formation. So inflammation of the muscle that leads to bone formation. That's what this condition literally is, and it is usually caused by trauma. So if you have some injury to a muscle, it can cause rupture of the muscle, and then eventually it can lead to metaplasia, which changes the muscle to bone. So that's the only reason you see a muscle shaped thing on x-ray. Usually muscles are like this on x-ray, but and bones are bright. But the reason that you're seeing this, which is like the shape of a muscle fiber, is that part has undergone ossification, which is metaplasia. Dysplasia is, I've also talked about it in between others, but it's not a true adaptive response. It's like uh, metaplasia and hyperplasia are the adaptive response, and then once the stress is too much, the mutations happen. If there's too much mutations, it can eventually lead to dysplasia, and dysplasia is the pre-malignant version, and it can, because it can go to cancer, but you have to know that dysplasia is reversible. So it's characterized by disordered growth of epithelium. There are a number of cellular changes that you can see, and these 4 points are really important to keep in mind, because they'll just give you a histology slide and they will say, okay, what is the characteristic of dysplasia that you see in this slide. So definitely you need to know there's abnormally frequent mitotic figures. Okay, so the main idea to remember all 4 points is that you have to know that there is too many mutations that leads to excessive cell division. So if there's excessive cell division, that you know that nucleus, there's DNA, and DNA has to replicate. So for the DNA to replicate and the cells to replicate, you need to go mitosis. You need to do mitosis. So in these cases, you will definitely see mitotic figures frequent, more frequent than normal, and pleomorphism. Each cell will have a different shape. So usually cells, because of their cytoskeleton, maintain a normal cell shape, but they can have increased different cell shape, because now you have different mutations in different parts of the cell. There's an increased nuclear to cytoplasmic ratio. So usually nucleus is in the center, there's like a 50% of like cytoplasm, and then the cell membrane. But what happens in these dysplastic cells is, because of accumulation of mutations and the DNA is always uncoiled, the nucleus is much bigger than a normal cell. So almost nucleus might take up almost 50, not 50, sorry, 70 to 80% of the whole cell's volume. That's also something you can look out for. And the chromatin, which is the genetic material, might be clumped in there. And this can be reversed. So if the stressor is removed, mild to moderate dysplasia uh, can regress, and it can change. And but sometimes severe dysplasia, which has affected like most of your stem cells, it can develop into carcinoma. A very important example of this you can see in cervical cancer. So cervical cancer is caused by human papilloma virus. So in the cervix of the uterus, you have different cell types. So this is the stratified squamous that we were talking about before. This is the normal. So this is the stem cell lining with the basement membrane. As you can see, the cells eventually, when these cells divide, they'll push each other, and you'll form the normal stratified layer. What happens is there's a mutation in the cell, in the stem cells, which can cause changes in the other cell, other cells, telling cells a lot. What I meant is, as you can see, the nuclear material has increased. There's more pleomorphism. Pleomorphism meaning cell shapes are much more varied, and the nucleus is much darker because of increased chromatin material. If you go next one more step further, meaning more mutations, you can see that there's no basement membrane. There's increased ratio of nucleus to cytoplasm, and there's mitotic figures. You can see some clumping of chromatin here. These are mitotic figures, like separating chromatin. And eventually cancer is when these abnormal cells not only divide within them, but break through this basement membrane, so it's invasive. So once they invade this part, that's when you know it's an invasive cancer. Yeah, I'm just not gonna go through this, but this was a drawing. This is like a actual histology slide. So this is normal, and then once it reaches like full thickness dysplasia, you can see like the cells are supposed to be like mostly pink, but you can see only nuclear material here. So just keep that in mind. Another example of change, so you can see like normal epithelium here, and here is dysplastic epithelium because of the, look at, like it's really chaotic and disorganized. Like at least here you can see some organization, but here the basement membrane is gone, and there's disorganization of there's increased nuclear to cytoplasm ratio, there's chromatin clumping and other things. Aplasia and hyperplasia, I just wanted to put here just so that you guys, when you see it, you don't think what they are. Aplasia means there is a failure of cell production and during embryogenesis, so that thing does not develop at all. Uh, if they don't get the right signals, so aplasia means no development. Hypoplasia is less development. So either there was not enough stimulus for an organ to develop, or if yeah, there's not enough stimulus during embryogenesis for an organ to develop. So example is a streak ovary in Turner syndrome. So Turner syndrome patients, their ovaries are hypoplastic, meaning they don't function that much, so they don't produce that much estrogen. So that's one of the problems in Turner syndrome. This is just a summary of everything: hyperplasia, hypertrophy, atrophy, dysplasia, metaplasia. So do you guys have any question on growth adaptations till now, or any suggestions or anything that you have, if you might know something that might help others, anything? All right, then if you guys do not have any the suggestions, we'll move on to Ashwin. He will be talking about irreversible and reversible injury. Do you guys want to break in between now, or
Yes,
How
I
about
want to say.
you give them a question and then we go for a
okay,
break?
fine. Do you guys want to do a question from you world before we go to the break?
Yeah.
It'll probably take just 5 minutes. Yeah.
All right. uh, 1 second, let me just load up the question. All right, can you guys see the question? So usually one question like this, you'll have 90 seconds to do it. So we can do it in two ways. One, we can have one of you guys read the question and then answer it, and then we'll discuss the answers. Or we can have everyone, maybe after around 90 seconds, just type in your answer in the chat. Everyone has to give in the answer, and then we can discuss the various answer choices and which is right and which is wrong. And this is from something that we covered today. So which one do you guys prefer?
Second one is nice option.
Okay, no worries. So you guys can go ahead and read the question, and I'll tell you when the time's up. Okay, guys, do you have an answer by now, or do you need more time? All right. All right, guys. So, what answers does everyone have? Just type it in the chat box, the letter. Okay.
Yeah, everybody just give it a go. It's alright if you're
Yeah.
wrong.
We'll go through the question together and discuss each option.
Expecting, like, I guess, three more answers and then, yeah.
Okay, so let's look at this question together and then we'll go through the answer options. So, first, 62-year-old man, worsening cough, shortness of breath, hemoptysis for the past 6 months. He has weight loss. So, he has this much 15 kilogram weight loss over 6 months. He definitely probably has some kind of cancer that we can think about, and with like coughing, shortness of breath, and hemoptysis is coughing out blood, so that is also something you might have to know. Is And 15 kilogram weight loss with 50 pack year smoking history, he most likely has cancer. So for a 62-year-old man to have a BMI of 18 is pretty low. That I'm sure you guys caught that as well. The next thing you have to keep in mind is the physical examination finding. They say that patient appears cachectic. So cachectic means there's a lot of muscle wasting, and with temporal wasting. Temporal wasting means that the muscle next to your ear, that right on top of your ear in the temporal region of your skull, the muscle on top, which is called temporalis, is on both the sides usually get wasted in cancer patients for some reason. So there's a loss of muscle mass over there, and there's generalized loss of muscle mass, meaning every other part of his body as well, his muscles have decreased. Chest x-ray shows a large lung mass, so now we know he probably has lung cancer. And he has some pleural effusion, meaning fluid in between the pleura of the lungs. So, which of these following cellular processes is most likely responsible for this patient's muscle loss? So, one thing you can do, approach to this question, is you can think of the answer yourself before reading the answer choices, because that might be a distraction, but it depends on however you want to do it. So, if we think about it the logical way, this man has cancer and he has weight loss, and the muscles have decreased, so there's generalized loss of muscle. So we saw before that skeletal muscles are permanent cells, so permanent cells, they cannot decrease in cell number, so skeletal muscles can only decrease in cell size. So for cell size to decrease, what is that pathway that we saw is needed to happen, that Nobel Prize winning pathway I mentioned before? So that is ubiquitin proteasome degradation. You need to decrease the amount of organelles and proteins in the cell to reduce the cell size, because skeletal muscles cannot undergo a decrease in cell number, so they undergo a decrease in cell size using the ubiquitin proteasome degradation pathway. You can think about it that way and then look at the answer choices. It might be a bit more clear. So covalent attachment of ubiquitin to muscle proteins. So if we remember that slide that I showed you, that how do cells degrade proteins? They attach with ubiquitin ligase. They attach the ubiquitin molecule onto the protein. They basically just signal the cell that okay, this guy needs to be destroyed, and the proteasome sees it and destroys it. So the proteins go down, cell size will go down. So A is looking good right now. B, plasma membrane rupture with leakage of cellular contents. This is something you guys haven't studied yet, but Ashwin will touch upon it next. It's basically describing a process in apoptosis, I think. It's apoptosis. So
Necrosis.
necrosis. So these are the answer explanation that UWorld has given, which is absolutely gold. Uh, I'm sure you guys have realized it by now. It's probably ubiquitin protease or protein degradation, because I wanted you guys to understand the reasoning that UWorld gives behind the other options that you have chosen. But B, which most of you guys chose as well, is plasma membrane rupture. It happens in necrosis. Plasma membrane instability due to defective dystrophin. So dystrophin is a cellular structure proteins like cytoskeletal protein found in muscle cells, and defective dystrophin leads to muscular dystrophy. So like Duchenne muscular dystrophy or Becker muscular dystrophy. So these things you would have to know. So that's why if you don't know the answer, it might be better to come up with your own reasoning and then look at the answer choices, because the others can probably confuse you. We have progressive shortening of chromosomal telomeres that happens in over time, meaning with each replication there's a shortening of chromosomal telomeres, and uh, that would lead to mutations and dysplasia rather than actually causing decrease in cell size, which is what we are looking for. We are looking for atrophy. Reprogramming of undifferentiated mesenchymal cells. That reprogramming, we saw that definitely means metaplasia. Metaplasia is changing from one cell type, which in this case is undifferentiated mesenchymal cells. So mesenchymal cells are the cells that give rise to your skeletal muscles and connective tissue. So these cells, reprogramming of them can, there'll be a metaplastic change. So in this case, the best answer looks like covalent attachment of ubiquitin to muscle protein. It's fine that you guys did not get this one right, because you just have to get used to the way to approach a question. That has to do more with practice and how many questions you're exposed to, because it's a topic that you guys are familiar with as well now. So this is their diagram of how the pathway works. So there's a target protein. Ubiquitin gets added by that complex that I showed you, ubiquitin ligase, with the help of ATP, of course. So it gets tagged, polyubiquitinated, and then it gets degraded by the proteasome into peptides. Yeah, so this is exactly what they're saying. Cachexia it's a cancer related thing. It happens in cancer patients. The details we will look at later on. And so ubiquitin proteasome pathway is what they are saying to degrade skeletal muscle proteins, so like actin and myosin. Um, this pathway is the major eukaryotic mechanism for selective proteolysis. So I told you, since this pathway won a Nobel Prize, I'm pretty sure it might show up one way or another on this exam. And the addition of ubiquitin multimers, so all those ubiquitin molecules that gets tagged, marks the protein for its rapid destruction by the proteasome. And so what happens inside the proteasome is like the protein is unfolded, and it's chopped into small peptides, and it can again there are other enzymes that can further degrade it to individual amino acids. So the reason that they gave for the part B, which you guys chose, is necrosis, because you guys did not know it for now. Necrosis is characterized by cellular injury and premature death due to external factors such as infection, toxin, or trauma. irreparable damage. So once the cell membrane explodes, meaning it's a damage that cannot be taken back. It's an irreversible change. This is another. This is the pathway in which particles are processed. So how you can move pathways across the cell. Wait, sorry. Yeah, again, so dystrophin, it has to do with muscular cells only, muscle cells. Uh, Telomeres, again, they can lead to cancer, but it's very unlikely to lead to metaplasia. I mean, sorry, atrophy. And the last one, reprogramming. Anytime you hear the word reprogramming, it's better to think of metaplasia. Metaplasia is changing from one cell type to another. And educational objective, so you have to know, so main point of this question was to know that whenever there's protein degradation, there has to be ubiquitin proteasome pathway involved. And in skeletal muscles, which is a non-replicating cell, it cannot decrease in cell number, so it has to decrease in cell size, so it goes through this pathway. And this is another diagram that was in the question regarding squamous metaplasia. We saw this is the normal respiratory epithelium. This is the pseudostratified ciliated columna. And then because of smoking, it will change into stratified squamous epithelium. And if there's enough mutations, it will become dysplastic with like increased nuclear cytoplasm ratio, pleomorphism, and all that stuff. Yeah, so that's it for the question. So I think right now Ashwin will take over for the next topic.
Yeah, but before that, they can
Mhm.
take a breather, like 5 minutes or so.
Oh yeah, you guys can take a break of 5 minutes.
Don't leave the meeting, guys. Just
Yeah.
take a break and be back.
Hello. Yeah. What is up? Is it a break? Yeah, it's 5 minutes break. How long did it take? Almost in we started 10, so it's almost 10 we started, so almost 1 hour. This thing. Nice. Yeah. Only 8 people, no? Yeah, there are like 10 or 11 on the start, and then in between just a lot of people just went off. Yeah. Nothing. I'm waiting for the question break to finish. No, no, not question. One question we discussed together, and then now break, and then Ashwin will start the next topic. Yes. No, no, I won't ask from you specifically. I'll ask you if you know the answer or something. Yes. But like the first class is this. Gautam, all those guys are here. That's Ashwin. Yeah, he is brown. Yeah, we can. Yeah. Okay. All right. Okay. Yeah. Okay. 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 Bye.
Guys, are we good to go? Do we have everybody here?
Yeah.
Cool, then I'll start. You guys can see the slide, right?
Yes.
Yeah. So Abhi already talked about the different adaptable changes that the cell can undergo, but obviously there's a threshold. You can bend, but you can't break, right? So once you pass a threshold, obviously cell will undergo injury, and it can either be reversible and irreversible. Since we have people from all years in this class, I'll just touch upon the different cellular organelles and their function before I move ahead. So obviously, the powerhouse of the cell, everybody knows it by that name, is the mitochondria. It's important for ATP production. There's something called the electron transport, which creates a gradient. So basically, it's electricity, right? You need a difference in the amount of charge in both sides of a membrane that is not permeable or semi-permeable. So since there's a difference in the amount of charge, once it moves through the membrane, you create energy. That's how ADP becomes ATP. So a mitochondria is a powerhouse of the cell. Then we have the smooth and the rough endoplasmic reticulum. It's basically the last junction where the DNA converts to RNA, and then from RNA you get protein. So it's basically the part of the cell which is responsible for making protein. Then there are lysosomes, which helps in degradation of the cell. There's Golgi apparatus, which helps in movement. It transports stuff from the nucleus to the rough endoplasmic reticulum and vice versa to the plasma membrane as well. Microtubules, again part of the cytoskeleton, helps with movement. And ribosomes, they attach themselves to the rough endoplasmic reticulum, and they kind of act like enzymes, ribozymes, right? So they act as enzymes that help works as a catalyst for the reactions that end up in making proteins when RNA is turned into protein. So there's one more important aspect of the plasma membrane that we have to know before we touch on cell injury. So the plasma membrane, as I've already told, is a semi-permeable membrane, and there are a lot of openings. So plasma membrane is made up of phospholipids, right? There are lipids, and the polar ends are towards the corners, and the non-polar ends are towards the center. And there are so many openings in this membrane for ions to move through. I think Abhi had obviously mentioned in the beginning that there's a gradient and there are the ions are spread differently. there's extracellular space and intracellular space. It's very self-explanatory. Extracellular space is the one that is outside your cell, and intracellular is the one that is inside. In the components of the ions that are present in the extracellular and the intracellular cellular space are very different. For examples, let's take sodium and potassium, right? Sodium is abundant in the extracellular space, and potassium is abundant in the intracellular space. And if you remember the concept of osmosis, and basically anything like diffusion, before we talk about osmosis, let's talk about diffusion. Diffusion happens from something that is at a higher gradient to something to a low gradient. So if you have a lot of potassium on the outside and low potassium in the inside, potassium naturally will move from the outside to the inside because that's the gradient. Vice versa for I'm sorry, I was mentioning sodium, right? So if sodium is if you have a lot of sodium in the outside and less sodium to inside the cell, the natural gradient for the sodium to move will be from the outside to the inside. It's vice versa for potassium. Potassium is abundant inside the cell and less outside. So potassium will naturally move from the inside to the outside. But that since we have to maintain a gradient and we cannot have an equilibrium between the ions, like you cannot have 10 molecules of sodium inside and 10 molecules of sodium outside, we need to have a gradient for electrons to flow. There is a pathway by which we supply energy for these ions to move against their gradient. So as you can see in the first image, the left bottom corner, we have ATP that is attached to these ion channels, and we spend our energy. ATP turns into ADP in moving these ions against the gradient. So I had mentioned that sodium is abundant outside the cell. We spend energy to maintain that gradient by sending excess sodium, even more sodium from the inside to the outside. And in that process, we also get potassium into the cell, which is also against its personal gradient, right? Potassium is abundant in the inside, but we still spend energy to get potassium from the outside to inside. So what happens is we take ATP, attach it to the ion channel. We spend that energy, one phosphate bond is broken, and in that process, 3 molecules of sodium is moved for 2 molecules of potassium. 3 molecules of sodium goes outside and 2 molecules of potassium comes inside. This is how diffusion works. Instead of, so we are trying to fight the natural process by supplying energy and moving it in the opposite side of the gradient. Osmosis is the movement of water through a gradient. So suppose you have 10 grams of salt in one solution. You separate it with another solution that has 15 grams of salt. So one is more concentrated than the other, right? So water moves from substance that has. So water will try to equilibrate between the two. So salt cannot move through the membrane. Only water can move through the membrane. So the water will move from the membrane which has more salt to the one which has less salt, if I'm not wrong. Just give me a second to orient myself. I'm sorry. So in case of this scenario, what I just mentioned, where imagine there's a big bowl that's separated by a membrane, which is semi-permeable and it can only move water through that membrane. So you have 10 grams of sodium in one and you have 15 grams of sodium in the other. So since salt itself cannot move through the membrane, you can only move water through the membrane to equilibrate the both. So if you move more water from the 10 gram of salt, from the solution with 10 grams of salt to the one with 15, the concentration of salt and the 15 gram solution will decrease, right? And you'll probably equilibrate at 12.5 in both the solutions. That is the concept of osmosis. So one of the main functions of our membrane is plasma membrane is to remain semi-permeable and to maintain a gradient between the extracellular and the intracellular fluid. I just wanted to put in a clinical correlation with the drug digitalis. Some of you might have heard of it. It is used in heart failure and conditions like arrhythmias. So what happens is when your heart does not do a good job of pumping blood, we need to supplement it with drugs that help it pump more efficiently. And how digitalis works is it blocks the sodium-potassium channel. So when you block the sodium and potassium channel, as I mentioned, sodium goes out, 3 molecules of sodium goes out, and 2 molecules of potassium come in. When you block this channel, all the sodium builds up inside the cell and the potassium builds up outside the cell. Now, this is just part of the membrane, right? This one channel is just one of many channels that are present in the membrane. So there's one more channel which exchanges sodium for calcium. Both of these channels work simultaneously at the same time. I'm just dealing with them separately. So in this channel, when you block this channel, sodium builds up on the inside. There's another channel which is responsible for excreting calcium and bringing in sodium. The point of the drug digitalis is that once you block this channel and sodium builds up inside the cell, there is no point in the second transporter. As in, since the whole purpose of the second transporter of sodium and calcium is to bring sodium in. Once you've already achieved that by blocking the sodium's exit in the first channel, you do not have to bring sodium in through the second channel. So this channel becomes redundant, right? The one with the sodium and calcium, which works to bring sodium inside, is not needed anymore because sodium is being built up by blocking the initial channel. What happens then is that you do not have to excrete calcium. When you do not have to excrete calcium, calcium builds up in the inside. So when calcium builds up in the inside, some of you might be aware that how a muscle contracts, you need calcium for that process. So once you build calcium up in the inside, muscle contracts much more efficiently. That's how digitalis helps our muscle, cardiac muscle, function better and pump blood more efficiently. It's just an example of how the ion channels can be manipulated in the form of drugs to help us work better. So now I'll touch upon how reversible and irreversible injury happens. So like Abhi mentioned, there are two cell processes that can take place once a cell is under stress, right? One is adaptation and one is injury. The adaptation happens in the cell continues to be under stress, it'll undergo injury. Once injury happens, there are one of two ways that the cell can undergo cell death. One is necrosis and the other is apoptosis. In the question that you had gotten wrong, the second option that said that the plasma membrane ruptures and the intracellular organelles and enzymes move towards the outside was a description of necrosis. And we will be dealing with necrosis in this when we talk about cell injury initially, and then I'll talk about apoptosis on the upcoming slides. So the first change that happens, like there are a lot of stimuluses, like it was mentioned in the question. It can be de-innervation, it can be mechanical trauma, you can be. I'm sorry, one second. Yeah, so there are different stresses that can take place to the cell. It can be mechanical trauma, like you can experience some sort of an accident, or it can be de-innervation, or it could be some metabolic cause where you're not feeding yourself well, or it could be, like Abhi had mentioned, pressure ulcers where you're lying down on the bed for a long time and your cells are physically being crushed, or it can be cancer treatment, which is interesting because the way we treat cancer is because cancer cells divide faster than other cells in your body. So you give in drugs that stop division, but you expect these drugs to work better on cancer cells because they are dividing faster than the other cells in your body. But that doesn't mean they are very exclusive, that they select only the cells that are cancerous and attack them. That we are developing medicines for that, in which we can target cancer cells in specific. But in general, chemotherapy and radiation, they affect cells that are dividing fast, but they also affect other normal tissue that is also undergoing natural division, right? So all of these are stresses to your cells, and any of this can cause necrosis. So the first step of necrosis is always reversible, and then once that phase is done and you still do not recover, if you do recover, you can go back to being normal, right? The cell can undergo stress, reversible cell changes can take place, and from then on, the cell can recover and become normal. But if that doesn't happen, you would go ahead and develop irreversible changes. To talk about the reversible changes, first up, as soon as necrosis takes place, the first thing that will happen is disruption of the ion channels. So when these ATP-dependent ion channels don't work, the first thing that will happen is the ion balance is disturbed. More sodium comes in, and as I said, water follows from something that has a lower concentration to a higher concentration. So if there's less sodium outside and more sodium is building up in the inside, water will follow. And when water follows, there's an increase in volume. and the cell size increases. When cell size increases, everything inside the cell also swells up. So, as the plasma membrane and the mitochondria, which are also membrane-bound organelles, they will swell up. And once that happens, the endoplasmic. Once that happens, basically, if the mitochondria swells up, as I had mentioned, the ATP is produced in the electron transport chain, which is part of the mitochondrial membrane. So, if that swells up and it is not functioning properly, the depletion of ATP becomes worse. You have lesser and lesser energy. And when that happens, you cannot function well. If the function of the cell, let's say, is to contract, if it's a muscular cell, that you cannot, the muscular cells cannot contract without ATP, and they stop functioning. And then there is chromatin clumping and ribosomal and polysomal detachment. So I remember mentioning that SER and smooth and rough endoplasmic reticulum are important for for making protein, and ribosomes are a catalyst that help out in that reaction. So when these membranes swells up, the dots that you have seen on the ribosomal membrane, the only difference between smooth and rough are the dots that you can see on the ribosomal membrane. These dots refer to the ribosomes itself, and once the cell swells up, these ribosomes detach from the endoplasmic reticulum, and that's when your cell stops making protein. All these changes that I mentioned up until now, the swelling of the plasma membrane, the swelling of the mitochondria, and the detachment of the ribosomes from the endoplasmic reticulum, all of them are reversible changes. As in, once the stress stops, all these changes can be reversed, and the cell can become normal again. Irreversible changes, like the word mentions, are changes from which the cell cannot revert. As in, with any one of these changes taking place, it confirms the fact that the cell is going to die. There's no way back. It's the point beyond Plato. So the first thing that happens after all these reversible changes happens, the next thing that will take place is there are multiple ways that the cell can rupture. Right, there are multiple mechanisms in place because of which your cell can undergo necrosis. One of which is lysosomal rupture. When I was discussing the organelles, I had mentioned the function of a lysosome. So lysosomes are part of your cells which contain hydraulic enzymes which are really acidic. The cells, for them to be in homeostasis, there's a certain pH at which they can tolerate. So once your lysosome ruptures and there are hydrolysic enzymes that are released, these change the pH of the cell, and most of the organelles of the cell cannot function in a very, very low pH, in a very, very acidic pH. That will lead to the death of the cells. So lysosomal rupture is one of the pathways by which necrosis can take place. Secondly, there's an enzyme called cytochrome C, which is also part of the mitochondrial membrane. After the mitochondrial membrane swells, once it ruptures, like it's basically like a balloon. Once you pump air into it, it'll swell, and at one point it'll rupture. Once that rupture takes place, cytochrome C can be released uh, into the cytosol, the cytoplasm. Once cytochrome C is released into the cytoplasm, it can activate enzymes by itself, which can cause necrosis. And as I had mentioned, once the pH for the homeostasis is not maintained, a lot of enzymes that are responsible for the degradation of the cell is activated. That's what I meant by degradative enzyme activation. Once that takes place, you'll start eating up your own cell. Pycnosis, karyohexis, and karyolysis are what happens inside the nucleus. The nucleus contains chromosomes inside. Pycnosis basically means condensation of the chromosomes. For the chromosomes to take place, like there are two processes that take place inside the nucleus, right? DNA to DNA and DNA to RNA. For these processes to take place, you have to separate the chromosomes and then divide them. So usually in a cell that is actively dividing, there is active chromatin, and there's chromatin that is not so active and not being divided at that moment. But when necrosis takes place, all the chromatin condenses, which is the process. That process is named pycnosis. And then there's karyohexis, where there's fragmentation of the chromosome. So all these chromosomes are torn apart, and karyolysis, where they just get degraded, right? So this is one of the markers for cell death, when the chromosome first condenses, and then after it condenses, it is fragmented, and then it degrades itself. It is very, very important to differentiate between the two changes. In a question stem, if they mention the fact that the cells are undergoing swelling, you have to remember that the process taking place at that point in time is necrosis and not apoptosis. And secondly, when they mention that the cell is swelling, they might go into the specifics and mention that the plasma membrane or the mitochondria is swelling. That's when you need to understand, okay, it is not beyond the point of no return. There is a possibility that this cell can be reverted back to normal. But if they mention something like pycnosis, they wouldn't actually give you the term pycnosis because it's a giveaway. They would say something like, while looking into the slide, they saw that the nucleus has been fragmented, the chromosomes inside the nucleus has been fragmented. And that's your clue to understanding the fact that the cell is beyond repair and it is destined to die. Apoptosis is the process in which, so you got to differentiate between necrosis and apoptosis. Whenever necrosis takes place, it induces inflammation. What happens is, as soon as like one of the steps in this was plasma membrane rupture, right? And as soon as irreversible cell changes take place, the plasma membrane will rupture. With the rupture of plasma membrane, the stuff that are inside the cell are exposed to the outside. When they leak into the extracellular space, they can activate the immune system and induce inflammation. The major difference, if there's only one takeaway point that you can choose between necrosis and apoptosis, is the fact that necrosis will induce inflammation. Those changes will cause the immune system to be activated, and they'll try to fight the response. But in case of apoptosis, like Abhi had mentioned, is programmed cell death. It will not cause inflammation. And the reason for which I will be mentioning is because as soon as, so I'll mention how apoptosis takes place, but the final effect is that there's a whole cell which is membrane bound by the plasma membrane. Even after the cell undergoes apoptosis, it'll still be membrane bound, except now it is smaller molecular blebs. So the whole cell breaks apart, but even though it breaks apart, the membrane covers each and every small bleb. Because it's membrane bound, the organelles and the cytosol that is inside the cell doesn't leak to the outside. And because there's no leak, there's no trigger, and because there's no trigger, the immune system is not activated, and hence there won't be any inflammation. Since we are trying really hard to contain the inflammation, and since energy is being spent, this process is going to be ATP dependent. On the other hand, necrosis is not ATP dependent. That is also one of the important differences between apoptosis and necrosis. And as I'll be discussing in the next slide, there are two pathways by which apoptosis can take place. But both of these pathways end up in the activation of caspases. Caspases, if I were to expand, it's cysteine aspartase protease. So protease means AS is basically degradation. Protein is obviously is short for protein. So enzymes that degrade protein are proteases. As you know, we are all made up of 20 amino acids, and cysteine and aspartase are part of that. Caspases is short form for cysteine aspartase proteases, which means they break down protein. And since we are all made up of amino acids, it basically means it breaks down those amino acids. Both the intrinsic and the extrinsic pathways culminate in the same manner, as in they both activate caspases, which is responsible for killing the cell. Like I had already mentioned, the reason why inflammation doesn't take place with apoptosis is because although they break down into smaller bodies, all these bodies are membrane bound. So there is no leaking, and since there's no leaking, there's no activation. Another clue that you can see in question stems is that when you stain these, when you look into them with a microscope with the help of a stain, the cytoplasm is eosinophilic and the nucleus is basophilic. It's just something that you would encounter in a question where they could say that they looked into a slide and they saw a basophilic nuclei. It's just for you to, just so that you don't feel blank or intimidated by the fact that they mentioned that. I just wanted to put out there that if apoptosis is taking place, the cytoplasm is eosinophilic and the nucleus is basophilic. Another factor that that you will encounter is called DNA laddering. So when apoptosis breaks down the DNA, when fragmentation takes place, pycnosis, karyohexis, and karyolysis takes place, so the DNA is broken down in such a manner that they are either broken down into pairs of like, suppose you have I don't know, So 360 base pairs, they'll be broken into two exactly, and you'll have 18 in one hand and 18 in the other. So they'll be broken down in multiples of 180 base pairs. Base pairs is basically when there are four thymidine, guanines, A, T, G, and C, guanine, adenine, thymine, guanine, and cytosine. These are the four nucleotides that make up the DNA. And when one connects with the other, it's called a base pair. And what apoptosis does is that every time it takes place, it breaks down the DNA in pairs of 180. So there'll be 180 base pairs of DNA together, and then there's a break, and then there's 180 more. So DNA laddering is a process you would see on gel electrophoresis if apoptosis did take place. Another difference that I've not mentioned up until now is apoptosis usually takes place in very small group of cells, right? Either a singular cell can undergo apoptosis, or a very small group of cells can undergo apoptosis at the same time. But on the other hand, contrasting with necrosis, it usually takes place in large groups. That is also an important difference that you have to keep in your mind. So touching on the intrinsic and the extrinsic pathway, as I had mentioned, both of them culminate in activating caspases and thereby causing cell death. Two things can take place, intrinsic and extrinsic. I'll touch on extrinsic first. So in case of extrinsic pathway, your immune system is made up of adaptive immunity and passive immunity. In adaptive immunity, there's a cell called the T cell. Some of you may be familiar with it, some of you may not be. The only thing that you have to know about the T cell in this, for you to understand this concept, is that T cell is the one that gives the killer blow. Once you activate the immune system, there's a long pathway which culminates in the fact that the T cell will release enzymes, which is going to kill the cell that is being attacked or that is under danger. So the T cell will release enzymes like granzymes and perforins. These cause perforations in the plasma membrane. Like I had mentioned, plasma membrane is really important to maintain the ion difference between the inside and the outside. And once that is not being maintained, all sorts of osmosis, diffusion, and ion leakage takes place. And once that takes place, you can activate the caspases. Another pathway is called the FAS and the FAS ligand pathway. So FAS is again a receptor that are there on immune cells, and FAS ligand is the, let's say, like a lock and key mechanism. So you have a lock, and there's a cell that comes up with the key. So FAS is the key, and FAS ligand is the lock. Once it attaches, it kind of opens up a lock, and then that there are a lot of downstream processes that take place, and that also culminates in activating the caspases, which will lead to apoptosis. On the other hand, in the intrinsic pathway, so any kind of distress, denervation or starvation or ischemia, anything that activates the apoptotic pathway, there are substances like P53 and RB. All of these are present inside the nucleus, and they are responsible for controlling the fact, like Abhi had mentioned, the fidelity and the rate in which mutations take place, right? So we have multiple checkpoints in the cell cycle that will regulate the fact that if the mutation has taken place, either you try to amend the mutation by making changes to that small segment, or if it is beyond repair, you stop cell cycle and then you kill the cell. So the P53 is like a policeman for the cell cycle. It makes sure that it happens without any mutations. So once a mutation takes place, P53 pauses the cell cycle. Either it tries to repair it, or it activates the apoptotic pathway, and because of which you can end up killing the cell. So what happens in that case is all the stressors first activate P53, and through P53, the key event in all of the intrinsic apoptotic pathways is the activation of the mitochondrial membrane, by which cytochrome C is leaked into the cytoplasm. I had already mentioned this previously, I believe, when I was talking about different pathways. The fact that once cytochrome C is leaked into the cytoplasm, it can activate caspases. So we have mechanisms in place. We should be having mechanisms in place in the membrane that stops this process, right? and that is called the BCL2 family. The BCL2 family is basically a bunch of proteins which can either be pro-apoptotic, as in they favor the pathway of death, or they can be anti-apoptotic. They try to stop your cell from dying. Both of which are important at different points in time. If there is a mutation, then you have to kill the cell. If there is no mutation, you can't just keep leaking cytochrome C into the cytoplasm and activating apoptosis. So there are two proteins that you got to keep in mind, BAX and BAC. They both are pro-apoptotic proteins. If both of these proteins are activated in the membrane, they end up leaking cytochrome C and hence activating the pathway. On the other hand, there's a protein called BCL2, which is not written on the slide, but BCL2 by itself is anti-apoptotic, so it functions in a manner to stop cytochrome C from leaking through the mitochondrial membrane. Once cytochrome C leaks, or once the T cells or the fast ligand is activated, all of them culminate in activating caspase 8, which is an initiator caspases, right? Even in caspases, there are a lot of downstream processes that have to take place, and obviously caspases are named from, they're just numbered from 1 to 10 or more. And caspase 8, I think they're numbered by when they were discovered. It's not actually numbered by which one happens first, which caspase is activated first. That's why you can see that caspase 8 is activated first, and then it ends up with the executioner caspase, which is caspase 3. It was just numbered by when it was invented. So you invent caspase 1 first, but it's involved in a different step in the process. It's not the first step of the process, but you just discovered it first. So it's called caspase 1. So the number that that comes after caspase doesn't have anything to do with the order in which it is activated. It's just that caspase 8, you got to remember that caspase 8 is part of the initiator complex, and then it ends up with activating caspase 3, which is the executioner complex. So some more clinical correlations. So apoptosis can either be a good process in which you get rid of mutations, or it can be a process in which you don't want a cell to die, but because of inappropriate activation of the apoptosis, the cells end up dying. And on your slide are a few examples of that. Follicular lymphoma. So lymphoma is basically cancer of the lymph nodes. Lymph nodes are part of your immune system. They're present throughout your body, and if these lymph nodes are affected by cancer, it's called lymphoma. And follicular is one of the types of lymphoma. So lymphoma is usually divided into two types majorly based on histology, called Hodgkin's and non-Hodgkin's lymphoma. Within non-Hodgkin's lymphoma, follicular lymphoma is one of the most common types. And the reason I've brought this up in apoptosis is because in the previous slide I had mentioned BCL2, which is a protein that is anti-apoptotic, right? So it stops cytochrome C from leaking. So imagine you have a cell, there has been a mutation in the cell, but because BCL2 is really active, it is stopping cytochrome C from leaking into the cytoplasm, and hence you cannot kill the cell. So what happens when a cell that has gone rogue cannot be killed? It multiplies exponentially, and hence you get cancer. Cancer is basically a bunch of cells that is mutated and is going berserk. Usually cells that undergo replication have multiple mechanisms in place to control the rate at which they replicate. One of which is when a cell hits the boundary of another cell, it knows, okay, my region ends here and I need to stop replicating. And there are so many other ways in which it's called contact inhibition. When one cell comes in contact with another cell and realizes that it's time to, it's inappropriate to divide further, there's a lack of space, and hence they need to stop. And there are so many other ways in which we keep cell count under our control. But if you have a cell that is mutated, but you cannot stop it from dying because BCL2 is activated inappropriately, then it can lead to cancer. That's what happens in follicular lymphoma. We have a cell called a cell that codes for immunoglobulins, and a chromosome, right? So chromosome is what contains the code that will eventually give rise to protein and make immunoglobulins. So let's say plasma cells, which make immunoglobulins, which is again part of your immune system. So we have a chromosome which is responsible for making these immunoglobulins. What happens in follicular lymphoma is there's a translocation. Translocation basically means where you chop. It's basically like grafting, where you take, let's say you have chromosome 18 and chromosome 14, and every chromosome has different heads. So in the process of translocation, you chop off a little bit of this chromosome and it gets added to this. So when you say in case of follicular lymphoma, chromosome number 14 and chromosome number 18 undergo translocation. So they kind of chop some. So they get chopped. To put it bluntly, they get chopped, and parts of number 14 attaches with 18. What happens because of this translocation is that BCL2, which is anti-apoptotic, is inappropriately activated, because of which we cannot stop mutated cells from dying, and hence those cells proliferate, and it can lead to follicular lymphoma. So this is an example of how basic science can explain a phenomenon like cancer. Secondly, cervical cancer. Abhi had already mentioned cervical cancer, but he was talking about in the viewpoint of histology. But in cases of pathogenesis, HPV is called is a virus, and it's one of the again, I think it was a Nobel Prize discovering victory where they figured out that HPV is the one that is responsible, one of the viruses that could be responsible for causing cancer. And how it does it is very fascinating, if you say. Like it secretes two protein, E6 and E7. What it does is that I mentioned for apoptosis to be activated by the intrinsic pathway, P53 has to be upregulated, right? What this does is that it binds to P53 and it stops it from being activated. So once the policemen have been, let's say, shielded, there's nothing that can maintain law, and the cell again goes berserk. It starts dividing, proliferating without any control, and you can develop cancer. Cancer is basically when cell growth happens without any control, like uncontrollable cell growth of rogue cells is cancer. And how HPV does this is by inhibiting P53, which again prevents apoptosis and hence leads to uncontrollable cell growth. Thymus is again part of the immune system. As kids, we have a huge thymus, and then it slowly becomes smaller as we grow up. It is usually present in our throat. And so thymus is responsible for making T cells. T cells, as I mentioned before, have the power to secrete granzymes and perforins, which can cause cell death. So if they go rogue, if they do not have a mechanism by which they can realize which cells to attack and which cells to not attack, they can end up harming themselves, right? And this is a whole new sector called autoimmune diseases, where our own immune system let's say they're off target and they start attacking tissue that is part of our body, thinking that it is a foreign substance, right? That's the whole concept of autoimmune diseases. So the thymus has to be trained in such a manner that they recognize tissue that is normal, and they have a mechanism in place for them to differentiate between normal tissue and pathologic tissue, and then they can target their attack towards the pathologic tissue. So if so apoptosis is the process by which, and in specific it's the fast ligand process by which in the thymus, all the T cells that get attached to cells that are actually physiologic and are not, like, if they, so T cells have again a lock and key mechanism. They have different let's say ligands on their plasma membrane that is looking for places to attach, right? But if the T cells aberrantly get attached to cells that are normal, that shouldn't be attacked, then those T cells should undergo apoptosis. If they do not undergo apoptosis, these T cells will then proliferate and start attacking a wide range of tissue, which is important for our functioning, and it can lead to autoimmune diseases. So in the thymus, there is a mechanism, in specific fast ligand mechanism, where the T cells that react with tissue that is normal undergo apoptosis. If this process doesn't take place, these self-reacting T cells will be out into the system and they'll start attacking normal tissue, physiologic tissue, and it can lead to autoimmune diseases. One of the examples of which is autoimmune lymphoproliferative syndrome, the details of which you do not have to know now. You just have to know the mechanism. I just put it in there to make you understand how apoptosis can be useful and how it can be misused in the other examples. Even fingers, in the whole process of embryogenesis, we are just a big blob of tissue, right? And then we have to be carved. So the way we are carved is like imagine your hand. It was actually one big blob of tissue, and then apoptosis had to take place and remove all the excess tissue to shape your hands in the manner it is now. So it's rudimentary to think about it. Nobody actually thinks about how our fingers look the way it does. But yeah, apoptosis is responsible for that as well. Abhi, do you want to take over now? Yeah.
So, do you guys have any questions regarding the topics that Ashwin just discussed?
I'm sorry if I spoke really fast.
Um, so, what do you guys think? So, do you guys feel like you're understanding the material, or do you want any other changes how we discuss it? Okay. Yeah, I guess.
Pretty
Yeah.
understandable.
Okay. So, I'll just continue with where Ashwin left off.
Abhi, they can't hear you now.
Yeah. Uh, 1 second. Yeah. Okay. So, yeah, I'll talk about necrosis, which is another necrosis, which is another way that cells can undergo, which is another way that cells die. So necrosis, apoptosis, Ashwin just previously discussed. So the basic definition of necrosis is it's death of a large group of cells, and it is followed by acute inflammation. So this is a big difference between apoptosis and necrosis that you have to remember, is that necrosis, it is followed by acute inflammation. Apoptosis is just the cell, it's a programmed cell death, and there is no there's no cleanup. The acute inflammation is basically neutrophils and macrophages entering the place where the dead cells are and cleaning them up. But why it doesn't happen in apoptosis, you can understand, because you saw Ashwin mentioned something about the autophagy and autophagosome. So when they lyse autophagosomes, they lyse themselves, so they break down by themselves. They don't need the help of neutrophils like in necrosis, where it's just acute inflammation that can clean them up. So basic pathology behind necrosis is there's some kind of injury. It can be like trauma, or it can be lack of blood supply. So necrosis in the heart. So when a person has myocardial infarction or a heart attack, what happens is you lose the blood supply to the heart. Then that the part which is supplied by that artery, which is blocked, ends up dying. So it undergoes necrosis. And what that means is, what actually happens within the cell is the plasma membranes get damaged. So once the plasma membrane gets damaged, there's no barrier separating the extracellular and intracellular environment. So Ashwin was talking about how the sodium potassium pump is necessary to maintain a concentration gradient in order to produce ATP and maintain the normal cellular structure. So once the plasma membrane gets damaged, you cannot maintain a barrier between them, so there's no chance the cell will survive. But additional changes also do happen within the nucleus. It goes to pycnosis, karyohexosis, and karyolysis, which Ashwin, you talked about. Yes, I was out.
Yeah, Pycnosis.
Yeah, okay,
And yeah,
so those nuclear changes, which are a sign of irreversible injury, happen because necrosis is basically irreversible anyway. Then cell swelling, and once cell swelling happens, there's this membrane blebbing. So blebbing is like the plasma membrane loses its ability to maintain a circular or whatever structure shape it is, and these protrusions start forming, these blebs, and then they slowly detach from each other. And also within the cell, the organelles are meant for destruction. So the mitochondria degrades, the endoplasmic reticulum degrades, Golgi apparatus, all of them start degrading. Protein also gets denatured because now there's no barrier between the outside and the inside environment of the cell, so there can be acidity and all the different stressors that cause denaturation of the protein structure. And then eventually the cell is going to burst and leak of intracellular components. All these toxic components gets released: acid, hydrolytic enzymes, peptidases, all of those stuff, calcium ions gets released. And then this process triggers inflammation, because once these cellular components are released, they recruit macrophages, and macrophages and neutrophils get called into the site because of the concentration of all these toxic substances released from the cell. And then eventually these leukocytes, basically neutrophils, uh, end up cleaning the dead material. That's when you have pus forming, and eventually then there'll be reduction of the pus, and wound healing will happen. They'll place some scar tissue on top. Some fibrosis might happen. If it's a regeneratable area, then it will grow back again, things like that. Then there are different types of necrosis. There are the main different types of necrosis that you guys should be worried about is coagulative, liquefactive, caseous, fat, fibrinoid, and gangrenous. Yeah, I know there's a lot of different types, but the basic idea behind most of them is simple. That is cell death. Just These are categorized based on how they look from the outside. So if you see coagulative necrosis, you'll know the difference between that just based on looking at it. But the pathology also slightly differs between them. We'll look at them individually. So first we have coagulative necrosis. In coagulative necrosis, the main thing is, yes, there's cell death happening, but the architecture of that cell will be preserved. So once that death happens, uh, sorry, one second. Yeah, once that death happens, the architecture, so the framework will still be maintained. So basically the cell just coagulates, but the shape of that organ will be mostly intact. That's why it's called coagulative necrosis. I'll tell you why more it's called coagulative. There's one more nice reason. But mainly this type of necrosis will happen in ischemic conditions, or because of infarctions, because of like embolisms from uh, from the heart or anywhere. So what happens, ischemia, if you guys are not sure, is lack of blood supply to an organ. So when most organs have ischemia, they will undergo coagulative necrosis. So kidney can undergo coagulative necrosis, the liver can uh, undergo coagulative necrosis, but every organ except the brain. So again, another exception. That means you have to remember this clearly. So they might ask you a question regarding like what is the process that happens after there is a stroke in the brain. So they might ask, will it be coagulative necrosis, liquefactive, or anything? It definitely is not coagulative necrosis, just in like in other organs, because in other organs it's coagulative. In the brain, it's not. Just keep that in mind. The next one is enzyme denaturation. That is the main pathophysiological mechanism that happens in that is important to remember in coagulative necrosis. So what I mean is, so once you lose the blood supply to the organ, so let's think about it in terms of cell. If the cell is not getting oxygen, it's not producing ATP. So if there's no production of ATP, what happens is the cell membrane and all the processes happening inside the cell to maintain normal function cannot take place, because ATP is the energy currency. So once that is not there, all the enzymes that need ATP, all the cell's membrane proteins that need ATP get blocked. They don't they don't get destroyed. They just do not happen because there's no ATP right now. Once they get blocked, what happens is, because like Ashwin mentioned, the sodium potassium pumps and all the pumps that maintain the concentration of ions and hydrogen ions cannot maintain the proper concentration. So in turn, it what happens is there will be denaturation because of the acidic environment that develops within the cell. Another way the acidity, if you want to think how it develops, is if there's lack of blood supply, then there's lack of oxygen. If there's lack of oxygen to the cell, it won't undergo aerobic respiration. So it will go undergo anaerobic respiration. In anaerobic respiration, I'm sure you guys are aware, lactic acid is produced, and that would increase the like intracellular acidity, which will lead to protein denaturation. And once protein are denatured, they'll get clumpy. So you've probably seen it, like once you take an egg and once you heat it, egg is basically protein. Once they denature, they clump together. So that's why you get in a hard boiled egg the thick white part, which is albumin and other proteins clumping. So that's why this clumping or coagulation is where the name coagulative necrosis comes from. So the main pathology is there's no ATP, all proteins are blocked. Then the acidic anaerobic respiration happens. Um, Lactic acid builds up. Internal acidity causes protein denaturation, which leads to clumping, coagulation, and coagulative necrosis. But the cell itself will maintain its architecture. The proteins are the ones that just get clumped because of this, and there's a lack of nuclei because that's how the cell is dead anyway. um, Increasing binding of eosin. So I'm sure I mentioned that before, hematoxylin and eosin stain. It's a sorry it's a stain that binds, that is commonly used in histology. It's one of the most common stains. And eosin, what it does usually is it binds to uh, acidic things. So once there's denaturation of protein and all this, eosin binds to basic things. Once there's too much protein denaturation and all these materials in that cell, uh, amino acids have nitrogenous bases. I'm sure you'll remember from chemistry. Amino acids are made from nitrogen bases, and because they can act as bases, eosin binds to basic substances, and all of the eosin will show up as in light pink on the histology slide. So it is indicative of necrosis, like dead material, dead protein, and dead intracellular structures. So this is the gross pathology slide of a kidney. So as you can see, this is kidney has undergone coagulative necrosis. The necrosed material is white in color here. And one more important thing to notice is there's a wedge shape in this infarction. So this by the way happened because some kind of loss of blood supply to one part of the kidney. So how the reason why this wedge shape is important to keep in mind is because the blood supply to the kidney starts off at one point and then it branches off. So these branching off is the reason why that the blood supply is in a wedge shape. So if the branching off happened somewhere around here and then you block the artery over here, you get a wedge shape infarct to this whole area. If another artery was blocked around here, you'd probably get a wedge shape over here. That's the main thing. And coagulative necrosis, this is how it looks like on like to the naked eye. But the kidney will still probably function, but not this kidney though. This kidney looks gone. uh, yeah, this is the histology slide of necrosis. This is a glomerulus. This is so how we can not glomerulus, this is a section of a kidney. And what we basically are looking for is, look at this, can you guys see this properly? This is a cellular structure with round this thing. This is a glomerulus. So this is a normal glomerulus, and you can see there's some blue nuclei and all this stuff around here. That's a normal glomerulus, as opposed to this round structures. Very compare the number of nuclei over here. So there's no nuclei, it's just filled with pink stuff. So those pink stuff are the coagulatively necrosed material. So those dead proteins which bind the eosin. And also we mentioned that coagulative necrosis, you will not see nucleus. So you don't see the nucleus in these ones. So you can see this border, like you can see this part has died off mostly. This part has nuclei, so it hasn't died off. And it's kidney because of these round structures and tubules, uh, glomeruli and its renal tubules. That's how it goes. Then that's coagulative necrosis. The next one is liquefactive necrosis. So if in coagulative necrosis proteins get clumped together, in liquefactive necrosis those protein gets degraded. So Usually liquefactive necrosis, what happens is there's some kind of insult leads to tissue dying, and once the tissue dying once the tissue dies, there's protein and cellular degradation. Okay, so in coagulative necrosis, the proteins are clumped and they are denatured, but in this case they are actively by like someone, either proteases or some neutrophil invasion, someone comes and digests these protein. That's why these all this material gets liquefied. And there's obviously loss of normal architecture, because now something or some process is actively degrading those material. You can think of it like this, like coagulation is like, okay, the protein gets denatured, they clump, and then liquefactive is when something or someone degrades all those proteins and make it more liquefied. It's like mixing it up together. So brain, we mentioned, it doesn't do coagulative necrosis. The only organ, if it loses its blood supply, does not do coagulative necrosis. What it undergoes is liquefactive necrosis. So what happens in the brain is, once you have ischemia to the brain, meaning you lose blood supply to part of the brain, then what happens is the cell, undergoes the normal process of necrosis that we discussed before, and it will lose its nucleus and all that. But then we have some support structures in the brain, some microglial cells. Microglial is like the fancy name for macrophages that live in the brain. So macrophage, they are involved in acute inflammation. What they do is they come in, they eat up the dead material in the brain. So they eat up all the dead material, then end up forming some pus, and then it's liquefied. That's why it has the name liquefactive. And then eventually it just leaves an empty cavity over there, eventually in the brain. So if you have, it depends on how the process turns out, but mostly that's how it happens. Another process that does liquefactive necrosis is bacterial abscesses. So I'm sure you guys have seen or heard of an abscess. An abscess is basically a cavity that is filled with pus and bacterial infection. So bacterial infection and immune cells in it. So bacterial abscesses also, what happens is this neutrophils and macrophages, they wall off the bacteria or the organism inside. They produce a boundary, stop them inside, and then start killing them in that area. So that area is filled with not just bacteria, but also dead bacteria, dead white blood cells. So it's like the battlefield, and the zone, they want to contain it in the bacterial abscess. And within that, the death that happens is liquefactive necrosis. So all the processes that involve the immune cells, like the acute inflammatory versions, undergo liquefactive necrosis. So in histology, what you would see for liquefactive necrosis is early macrophage invasion and cellular debris. So like I mentioned, there will be death, and then macrophages, if it's just in the brain, it's called microglia. They would invade, they would clean up, they would start their cleanup process and fighting whatever, if there's infection, they'll start fighting, and there'll be cellular debris from the dead material. And eventually, later on, once the macrophage cleans up all the dead material, starts its healing process, the space, that the boundary that all these were happening, can be left empty, so they can become a cyst or a cavity. So this happens in quite a few infections and strokes as well. So here you can see this person had a stroke to the brain. This is the gross pathological slide of the brain. So there was some kind of ischemia or infarction to the brain that resulted in the formation. So here you'll see the liquefactive material inside, liquefactive necrosis. That's what happened over here. And eventually, once this heals off, what will happen is all this material will be cleaned out, and it'll just have an empty cystic space, a cavity in the brain. So that is liquefactive necrosis. This is the histology slide. So the difference is in coagulative necrosis, you were seeing a lot of pink stuff without nuclei. In this liquefactive necrosis, we mentioned that a lot of neutrophils come and attack. So basically, that's what you should be looking for. So the pink stuff you're seeing is dead material, and there's a lot of blue stuff. If you zoom into this area, it would probably look like this. All these blue stuff are immune cells, actually neutrophils, that are invading this area to clean up that material. So this area is filled with, this pink is the dead material, with the blue being the neutrophils coming in to clean up the area. Next, do you guys have any questions till now about liquefactive or coagulative necrosis? No? Okay. Do you?
No, I said no.
Okay. Yeah, next we have caseous necrosis. So caseous, when you think about the word caseous, you should think about something like a soft cottage cheese, looking like paneer or like tofu or something like, not tofu, but like, yeah, that kind of look. So caseous necrosis, basically, just by looking at it, they named it because it something died, but then it ended up looking like cottage cheese. So they called it caseous necrosis. This type of necrosis you'll mostly see in things that form granulomas. Again, this is a new term, and you'll learn about this later on. But what I'm gonna say is granuloma is basically um, this process that happens during inflammation is macrophages come, and whatever bacteria or something, they wall it off and form a surrounding layer, almost like how we discussed in forming of a cavity, like an abscess. Similar to that, this is on a cellular level. What happens is, macrophages wall off the microorganisms and the dead material, and they stop the spread of that infection. This kind of this kind of necrosis, you can see in fungal infections. So if you guys know any fungi like aspergillus, histoplasma, do you guys know any other fungi? So most systemic fungal infections, you can get caseous necrosis. So meaning the dead material in that. Suppose you have a lung fungal infection, the dead part of the lung will look like cottage cheese. I have some disgusting pictures a little bit on later on about them. Then tuberculosis, it's a bacterial infection, but it can also cause caseous necrosis. In fact, I think caseous necrosis was probably first found in a tuberculosis patient, because tuberculosis is one of the oldest diseases that we have. And nocardia is another microorganism. Don't worry about the names of these organisms. You will be going through each organism separately anyway, but just know that fungi, tuberculosis, and nocardia cause caseous necrosis. There's an easy way to remember the ones that cause caseous necrosis. If you think that cheese is made of fat, and things that have a lot of like lipid material in them or require lipid to survive. So tuberculosis, they need something called mycolic acid to survive. It's a type of fatty acid, and fungi also need a specific type of fat to survive. So this caseation is like something they also involve in to like help them survive. uh, the fat and the dead fat in them also clump together and end up forming this cottage cheese like appearance. So that's one way you can remember it. Then there's always the dead material surrounded by lymphocytes and macrophages, because again they just come to clean, and they are the ones who have walled off the infection anyway. Yeah, this is a lung, and this is the pleural surface. So this is a TB patient, and as you can see, this looks like cottage cheese. And yeah, this is the walled off cavity. So yeah, this is a tuberculous cavity. It's called a caseum because it has caseous necrosis now. So you can easily differentiate caseous necrosis from any other type just by looking at it. And this is how it looks under a microscope, in a microscope. So this is the dead material, and this is the granuloma, walled off area with like a lot of lymphocytes and macrophages just surrounding this area. This is the dead material, and you can see some macrophages have decided to join together and form something called a giant macrophage, giant cell. So this is the histology slide of what inside this area boundary would look like. And then we have fat necrosis. So fat necrosis is a special type of necrosis that happens in fatty tissue when the adipose tissue die off. So the causes of a fatty tissue, it can either be because of enzymes degrading the fat, or you have a traumatic injury to an area where you have a lot of fatty tissue. So the enzymes that break up fat, so fat is lipid, so enzymes that break it up, it would be called lipases. So they break down the triacylglycerols, which are the building blocks of fatty acids. I mean, fat, and then they break it down to the individual component fatty acids. Then what happens is these fatty acids, because they are negatively charged, they will combine with calcium, which is positively charged, and they will form like a complex. And this formation is called saponification, meaning soap formation. Actually, in chemistry, if you want to form soap, it's related to linking fatty acids with calcification. So you form this kind of soapy white chalky material if the fat is broken down by enzymes. On the other hand, even if you have trauma and once the fat in that area is broken off, what happens is there will be some enzymes surrounding in the blood vessels that will cause the triacylglycerols to convert to fatty acids, and then the whole process will happen again in the area that you had trauma as well because of other enzymes present. So in fatty necrosis, where you'll see the outline of the adipocyte, but there won't be any nuclei inside, just like in other necrotic material. And if you think that there are nuclei in here, it's because those are the immune cells that are infiltrating. So the nuclei that you see interspaced even in between the pink stuff, they are actually from the neutrophils and lymphocytes that have infiltrated. Maybe some survivors, but yeah. Uh, yeah, so fatty necrosis is very easy to identify on histology slide. I think I have a slide next. Because this fat saponification and calcium on an H&E stain looks dark blue. So the only thing that looks dark blue on this is usually the nuclei. But even this area would fatty necrosis would look dark blue. So this is a picture of fat necrosis, how it looks like in a person. So as you can see, this person has a lot of subcutaneous fat, and maybe due to some kind of high speed trauma, or I'm not sure what happened over here, but this person had undergone necrosis of the subcutaneous fat, and it has calcified and caused fat necrosis. You can also see on the shoulder. So this is probably like multiple traumas. And this is a mammograph. A mammograph is basically like an X-ray of the breasts, and what it shows is there's a densely bright object over here. So in X-rays, usually things with calcium end up being really bright, so like bones and stuff. So in this case, the reason why this structure is white is bright is because it's filled with calcium. So this person probably had something to do with fatty necrosis, because breast is a tissue with a lot of adipocytes. So what happens during, if there's a seatbelt injury, when women wear seat belts and they undergo a high speed motor vehicle accident, there's a chance that the breasts undergo trauma because of the seat belt, and that is a common cause of fatty necrosis, or like a sports injury as well, can cause the breast adipocytes to undergo saponification, then calcification, which is part of how the fatty necrosis works. And the adipocytes are obviously dead. That's why the necrosis is there. This is the histology slide again. So you see the outline of the fatty adipocytes, but there's like nothing, no content, and it's probably just calcified. Fibrinoid necrosis. So fibrinoid necrosis, just from the name, there's necrosis, meaning there's some kind of death of large group of cells, and there's fibrin involved. So fibrin is a type of protein that we have floating around. It's involved in hemostasis, meaning like to help us stop bleeding. So fibrinoid necrosis, there's necrotic damage to blood vessel walls. So this type of necrosis, you'll only see it in blood vessels, and the reasons can be various, but mainly what happens is because of some kind of leakage that leads to some kind of leakage that happens. There's protein deposition along the blood vessel wall, which again causes more inflammation, acute inflammation. Once the protein is in a place where it should not be, then the body reacts to it, and then there's like acute inflammation. You'll have neutrophils as well entering there, but the main problem is there's protein deposits along the wall, and then there'll be necrosis because of the presence of protein in there. So yeah, the mechanism protein deposition, it can either be because of an immune complex reaction. We have something called vasculitis. Vasculitis is a condition in which there's inflammation of your blood vessels. So how and it's usually because your body is just attacking itself. So in that case there'll be antibodies against your blood vessels, and those antibodies end up, and antibodies are protein as well, yeah. So these antibodies bind to the blood vessel wall, and these proteins get deposited in the blood vessel wall, causing necrosis later on. Necrosis because inflammation follows, and eventually necrosis. Leakage of protein from vessels in malignant hypertension. So another way this can happen is just logically, best way this can happen is the blood vessel is like a tube made with like patch of cells. So if there's extremely high blood pressure within the vessel, what happens is it can cause stretching and eventually leakage. So once the cells are separated because of the high pressure, what happens is fibrin and many other proteins get deposited in between the cells, and this eventually leads to acute inflammation and then necrosis. So this thing happening in around a vessel wall is called fibrinoid necrosis, because fibrin is the main trigger for the inflammatory process. So it's probably why it's called fibrinoid necrosis. This is the histology slide of this. Again, necrosis, yellow material. So you can yellow material, pink or red material, pink material over here, and this is kind of different from the red and pink, like kind of reddish pink over here. So can anyone take a guess what these things are?
Blood cells?
Yes, exactly, because this is a blood vessel wall. This is the blood vessel, maybe an artery because of the thickness, a small artery, and this is the red blood cells that they just got caught up during the sectioning. And you can see there is this area where there's red or pink deposits over here without nuclei, and this is the proteinaceous deposits along the blood vessel wall. And eventually even the blood vessel has, like, lost its normal architecture, and there's like necrotic material everywhere. So this blood vessel is probably not, obviously not functioning. Then there's another type of necrosis called gangrenous necrosis. So I'm sure you guys have all heard of gangrene, which is like a dead, like when you think of gangrene you'd think of like a foot ulcer or a diabetic foot ulcer. Then that's exactly what gangrene is. It's a subtype of coagulative necrosis though. So let me remind you what coagulative necrosis is again. From just from the name, is there's some kind of coagulation happening and there's death of large group of cells. So ischemia, there's no ATP, anaerobic respiration, acidity, denaturation of protein. Once the proteins are denatured, they start clumping together, and that's why there's coagulation. So that is what coagulative necrosis mean, and gangrenous is just a subtype of it when it happens in your distal extremities, maybe in your fingers or legs, due to like blood vessel blockage, or it can also happen in your GI tract, your intestines, due to chronic ischemia. So if you look at it, just if you look at a gangrene, you would see mummified tissue. Like you would see it dead, but like it will look like a tissue that's mummified. And there's two different types: dry gangrene, wet gangrene. So first, dry gangrene forms, and when there's an infection on it, then neutrophils and immune cells enter and start working on the infection, and this causes a liquidy pussy area to form, and that's why it's called a wet gangrene, because neutrophils and there's degradation. And again, gangrene is like a mix of coagulative and liquefactive, because dry gangrene is just coagulative, and once the infection happens and neutrophils are called to the area, then neutrophils will clean up the dead tissue a bit and liquefy that area pus. So it's a liquefactive necrosis. So it's a combination of both of them. This is a dry gangrene. This is a dry gangrenous foot, probably from chronic ischemia to the leg. So this patient probably had atherosclerosis of the leg and eventually lost blood supply and ended up at this stage. But this is like a really severe one. You probably won't see it very commonly. This is a wet gangrene. So this is a foot ulcer, probably in a diabetic patient, and it's wet because it's undergoing liquefactive necrosis. It is probably infected. Next we have ischemia. So before we move on to ischemia, do you guys have any questions on necrosis, different types of necrosis? So we covered many different types and why they happen. So any questions?
No.
Do you guys want to break now, or do you want to continue?
Yes, break would be good.
Okay, so we'll have a 5 minutes break.
Break. 5 minutes. Oh, 1-1. Yeah, you can tell the others. What happened? It's a USMLE match. Yeah. Holy fuck. You playing in it? Yeah. Bro. Sahil was there. Huh? Sahil played some Chutia team, dude.
Sahil's team you played?
Yeah. Where is he? He was in Daisy, dude. Oh, how many people joined, right? Hmm? How many people joined? How many? 3. What? Three confirmed joined, paying the money?
I don't know.
He's last week, right? No, the basically
can we start, guys?
Yeah.
Yeah.
Thanks, man. Yeah. So we already talked about different mechanisms in which necrosis and apoptosis can take place. One of which is ischemia. Ischemia basically means decrease in blood supply to that organ. It can happen one of two ways. Either, so obviously, when there's an organ, there's an artery supplying blood to the organ, there's a vein taking blood away from the organ, right? So you can have stuff that decreases flow into the organ through the artery, things like atherosclerosis, where you have lipid that builds up in your vein, in your arteries, because of which the amount of area through which the blood can pass through decreases, and there's decreased blood to the organ. Or you can have something like a venous drainage issue, in which case there can be something that blocks the veins, because of which there's backward pressure, and the blood that is already devoid of oxygen. So your organ has taken up all the oxygen and the nutrients that it's needed, right? So this blood is blue blood, bad blood, or it doesn't have anything that the organ needs. But when this starts backing up because of the back pressure, it decreases the flow of blood which is rich with nutrients and oxygen into the organ. So either a problem with drainage or a problem with supply. Both of this can lead to ischemia. One of the important concepts in terms of anatomy that is tested with ischemia is called watershed region. Watershed region is basically parts of your body that have end organ supply that are at the borders. So what happens is in a lot of organs you have multiple arteries supplying that particular tissue, right? So if one goes haywire, the other can take over, and that can supply that particular region. But in certain parts of your body, there's only one artery, or there's only one source from which the blood can come. So already these parts of your body are more vulnerable than the others to ischemia. So now, when this particular region is towards suppose you have a blood vessel that is supplying a lot of tissues, right, from the start to the end. Let's take the example of the colon, which is the best way to explain this. Blood supply for the colon comes from the superior mesenteric artery and the inferior mesenteric artery. Superior mesenteric artery supplies the ascending colon and part of the transverse colon. Inferior mesenteric artery starts its blood supply from the descending colon towards up till the rectum. So splenic flexure, right, the place in the right corner or towards the top, if you guys can see it, that is the area where the supply of superior mesenteric ends and the inferior mesenteric starts. So that is the far end of the supply, right, for the superior SMA artery. Let's just call it SMA for ease. So SMA and IMA. So the area where SMA ends and IMA starts is the splenic flexure. So this area is easily prone to ischemia because it's at the far end of SMA and is just the starting of IMA. So, and it doesn't have any other collateral blood supply, which is any other artery supplying it. So it is extremely prone to ischemia. So watershed regions are those regions which are at the end of the blood supply of a certain artery, which doesn't have any collateral artery to supply it, which doesn't have any other artery supplying it. And there are different parts of every organ in our body that are prone to developing ischemia because they are in the watershed region. What I just explained is for the colon. In the colon, because the SMA finishes towards, I think it's 2/3 of the transverse colon. After that is where the SMA supply stops. So that region, because it's towards the end of the supply, is vulnerable. And the second region is the rectum, because that's where the supply of the IMA ends and the next artery begins. So that is also a region, it's like a junction where one ends and the other starts. And that is also prone to ischemia because damage to either one of them can affect that area. So any area where a blood supply stops and starts is prone to ischemia. Now let's take the example of a brain, which is in the bottom left corner. Brain is supplied by 3 arteries: anterior cerebral, middle cerebral, and posterior cerebral artery. And just like the colon, there are junctions where the anterior stops and the middle begins. And there are junctions where the middle stops and the posterior begins. The 2 areas that are highlighted with in violet, those are the junctions between the anterior and the middle, and the middle and the posterior. These regions are again prone to ischemia. And when you develop ischemia in regions like this, you develop a syndrome called man in a barrel syndrome. So you have to be familiar with which part of the brain is responsible for the movement of which part of your body and sensation in which part of your body to understand that syndrome. But just keep in your mind, it's called man in a barrel syndrome because your proximal extremities, which so the part of the brain that is affected by this syndrome is responsible for the movement and sensation of your extremities, proximal extremities. So if you cannot move your proximal extremities, you're kind of like that. So it feels like you're in a barrel. Barrel is a container and you cannot move. So it's called man in a barrel syndrome because you cannot move your extremities because the part of the brain that is responsible for that motion is lacking blood supply. The second example is that of the heart. So this is also a very good clinical correlation because it obviously stems into the fact of myocardial infarction, which is the number one killer even today. So the supply for although heart does a lot of pumping, it can't just diffuse blood. There's a small amount of blood that will diffuse through the membranes of the heart and supply blood to the heart itself, because heart in itself is also an organ that needs oxygen and nutrients for its survival. In fact, it needs a lot of it because it's doing a great amount of work and it exerts a lot of pressure in pumping blood, right? So it takes up a good amount of oxygen nutrients to do that. So in itself, the supply for blood for the heart starts from, there are three layers to the heart. Starting from the outside is the epicardium, and inside is the endocardium. And in the middle, you have muscle tissue. So the supply for the heart lies in the outside, which is the epicardium. So basically, there is blood vessels like this in the epicardium, and there are branches that get into the heart and try to supply. But the thickness of the heart, let's say, is from here to here, right? This is the epicardium and this is the endocardium. And there are branches that go in like this and supply the heart. But like we had mentioned in the first slide, there's if hypertrophy of the heart takes place in case of hypertension, this thickness increases. So instead of it being this thick, now it's this thick, right? And the blood supply is not sufficient enough to go through the whole depth and supply the whole heart, all the layers of the heart. That is when the innermost layer of the heart experiences ischemia first and it moves outward. So when you start learning about MI, or if you have learned about MI, you know there are two types of MI, right? ST segment elevated MI and non-ST segment elevated MI. These are basically changes in the ECG, the electronic reading of the heart's activity. But what it signifies is that when there is an non-ST segment elevated MI, the ischemia is contained within the innermost part of the heart. And the outermost region, which is where the blood supply comes from, the blood still manages to supply that area, but it is not great enough to supply the innermost regions. An ST segment elevated MI is when all the layers of the heart is affected and all of them are undergoing ischemia, because the amount of blood lost is much greater than a non-ST segment elevated MI, and it is not sufficient to supply any of the layers. That's when you get it's called an ST segment elevated MI. How the ECG works is for a different class, but this is the physiologic reason why those two are differentiated in such a manner. And so to the right bottom, you have, it's a liver, and it shows where the artery is and where the vein is. So if I were to ask you which zone, oh, sorry, myocardial infarction, that is when like a heart attack. So basically, that's when blood supply to the heart is blocked either by atherosclerosis, like we had mentioned, lipid buildup in the vessel, and the amount of blood flowing into that vessel decreases, and blood supply to the vessel decreases because of that. And it can happen to any organ, and if it happens to the heart, it's called myocardial infarction. Does that answer the question?
Yeah.
Yeah, cool. Thanks, man. Yeah, so to the bottom right corner, there's a picture of a segment of the liver. You can see where the artery begins and you can see where the vein is. So by pure logic, can any one of you tell me which zone will be affected first if ischemia sets in? You'll have to tell the answer out loud because I cannot see the Google Meet screen. So if any one of you know the answer, please tell it out loud.
Third zone.
Perfect. Yeah. that is the farthest away from the hepatic artery, right? So when there is ischemic hepatitis, which is basically inflammation of the liver because of ischemia, zone 3 is the one that is affected first, which is also a question. And it's really easy to understand the concept behind it and understand why that takes place. And I've also so I've mentioned about the neurons, the place of the brain that is affected by the blood supply, by a lack of blood supply, the watershed regions. But there are also some specific neurons. Neurons are the cells in your brain, right? There are some specific neurons that are more vulnerable to ischemia. It could be because of their anatomic location or because of their function, because every neuron will have a certain work rate, right? The amount of energy and the blood supply that they need to function. It can vary depending on what their function in the body is. So depending on that, some neurons may be more vulnerable to ischemia than the other. So you have to remember, for the sake of questions, that Purkinje cells in the cerebellum. Cerebellum is behind at the posterior part of your brain, and it is responsible for coordinating your motion and giving you balance. And there is a certain type of cell called the Purkinje cells, which is vital for the functioning of the cerebellum. And those cells, along with the pyramidal cells in the hippocampus. Hippocampus, as some of you may know, is important for memory. It is one of the regions of the brain that is affected in Alzheimer's. So it's important for recalling and memory storage. And there are cells called pyramidal cells in the hippocampus. So you have to know that the pyramidal cells of the hippocampus and the Purkinje cells of the cerebellum are extremely prone to ischemia. Thirdly, also the neocortex. The neocortex is the biggest part of your brain, and that is where all the areas of control of your sensation, your motor control, your speech, and all of that is located. You have to know that the third, fifth, and the sixth layers of the neocortex are also the most vulnerable. In the kidneys, which I have not, the diagram for which is not given here, which is why I put it up in text, is that the straight segment of the, so if you know the anatomy of the kidney, there's a glomerulus, and then there are tubules which take out and filter your urine. And initially when you start off, there's a lot of urine produced, and then from there on you reabsorb a little bit, you secrete a little bit, and there's the end product which you pee out. So there is a whole pathway from the glomerulus to the collecting ductule, which is the last part of the nephron. And like neuron is the cell in your brain, nephron is the cell in your kidneys. So there's a whole set of tubules that function to filter and secrete stuff into your urine before you pee it out. So one segment is called the straight segment of the proximal tubule, which is one of the first regions after the glomerulus. And the other one is called the thick ascending limb of the kidney. Both of this are present in the medulla of the kidney. Like all the other examples given, medulla is the innermost region of the nephron in the kidneys. So again, they are getting lesser blood supply, and the function they have in the proximal tubule, where they're reabsorbing a lot of stuff which requires a lot of energy, has a high work rate. So when again there's a difference in supply and demand, that's when ischemia sets in. And you have to remember these regions. You have to remember the splenic flexure and the colon. You have to remember the endocardium in the heart, zone 3 in the liver, and the straight segment and the ascending limb in the kidney. So moving on. Free radical damage. This is another concept that we'll be touching upon. So basically, in chemistry, some of you may recall that all the atoms, all the elements have paired electrons in their orbit. So every electron has a different spin, and they need to be paired in every suborbit of an atom. So if these electrons are not paired, they're highly reactive. They're either you lose that electron and you have a paired electron in your outermost complex, or you gain an electron from some other atom and you thereby complete your pair. Either you lose the one you have and complete your pair, or you get another one and complete your pair. Either way, they have to react with something, right? So they are extremely reactive. And this can be very corrosive, not just for us, but for the bacteria as well, or any organism that is living that attacks us. So what happens is all our immune cells try to make these free radicals so that we can attack bacteria. It's part of a process by which immune cells attack our bacteria. But again, we can have collateral damage. Like in that process by which the immune cells can attack bacteria, we can also experience damage to the cells of our body. So again, there have to be processes in place which try to regulate this and try to nullify the effect of those free radicals on our body, at the same time affecting the bacteria. So there has to be a balance. So to the right, there's a diagram which is gold. Like it's really important it's really important that you understand this because there are multiple clinical correlations. The first step is oxygen being converted to something called a superoxide. So basically, oxygen has gained an atom now in the process by which NADPH has lost one. So by oxygen gaining an atom, now it has an unpaired electron in its final orbit. It's extremely unstable, which can be which can be dangerous to both us and the organism that is invading us. So we have another enzyme called superoxide dismutase, the reaction for which is given in the left side, where oxygen takes up electrons from hydrogen and becomes H2O2, which is a much more stable molecule, and it is not as corrosive, right? So it is one of our defense mechanisms. That's why it's called scavenging, and that's why it's called scavenging enzymes, where it takes up the superoxide and gives you H2O2, which is much more stable and less harmful to us. Next up, there's another enzyme which is called myeloperoxidase, which converts H2O2 to HOCl, which is extremely corrosive for the bacteria, and hence you end up killing the bacteria. It's one of the mechanisms by how enzymes, like I mentioned initially, when lysosomes degrade the cell, they make blebs where they release enzymes, create an acidic environment, and release enzymes like these, which degrade whatever's inside that bleb. So once you corner the bacteria, make a bleb around it, and release myeloperoxidase and form HOCl, it can degrade the bacteria. Again, we have other mechanisms by which we have to support this reaction. So we have to keep supplying the NADPH, right? So once this reaction takes place, you're left with NADP+. You have to recycle it back to NADPH, for which we need an enzyme called G6P. Can you see number 6? Right, it's called G6PD, which is an enzyme. It's a whole chain of reaction which takes place for us to regenerate the NADPH to fight the molecule. So in summary, initially we have oxygen. We have an enzyme called NADPH oxidase, which converts the oxygen to superoxide. And then we have an enzyme called myeloperoxidase, which converts the final product to HOCl, which degrades the bacteria. Now, there are two clinical correlations that I'd like to talk about. First one is if our body lacks NADPH oxidase. So if you lack NADPH oxidase, you cannot convert the oxygen to superoxide. If that doesn't take place, everything below that also doesn't take place, and hence you don't end up killing the bacteria. Now there's a catch. Luckily, some of the bacteria that attack us provide us with H2O2. If they provide us with H2O2, we can bypass the step. Although our body lacks NADPH, since the bacteria itself is providing us with H2O2, we can use our myeloperoxidase and create HOCl and kill the bacteria. So we won't be in as much trouble as we imagined. But there are a few bacteria that can change the H2O2 that we have into water, which is H2O and O2. If that happens, we lose a substrate that is important for us to make HOCl. And when we lose that substrate, we cannot kill the bacteria. So in a disease in which you do not have NADPH oxidase, if a bacteria which has the enzyme catalase that converts H2O to H2O, you will not have the substrate to kill that bacteria. And hence, in this disease, all the bacteria with catalase will affect us and we won't be able to fight it. The prime example for such a bacteria, which is important for you to remember, is Staphylococcus aureus. Some of you may have come across it. It is the most abundantly discussed bacteria, one of the most abundantly, if not the most. and that bacteria has catalase, because of which it can change H2O2 to H2O, and hence we will not be forming HOCl, which will kill the bacteria. That is the first clinical correlation. Have you understood this disease? It's called chronic granulomatous disease, in which we lack NADPH oxidase. Guys, you have to, again, since I'm not on the screen, if you speak up, it'll be better for me. It's a difficult concept, so I'm ready to go through that again if you feel you've not understood it.
Yes, again, please.
Okay.
Thank you.
Cool. So we have mechanisms in place which first generate free radicals and then try to scavenge them. The ones that generate free radicals are stuff like NADPH oxidase and HOCl, myeloperoxidase, which will create highly reactive enzymes that can degrade the bacteria. The stuff that help us neutralize it is superoxide dismutase and G6PD, which helps in regenerating NADPH. So there are two sides to that coin. Now, have you got that? That part is clear, right?
Yeah.
Yes.
Yeah. So once you get that, now you need to realize what will happen if you do not have NADPH oxidase. That is the first step in the reaction. When you do not have NADPH oxidase, you cannot create superoxide. And without that, you cannot do anything that goes below that. So when you cannot create superoxide, you'll end up inefficient to kill a lot of bacteria. But to our luck, a lot of bacteria provide us with H2O2. Once they provide us with H2O2, we can bypass the NAD step that requires NADPH oxidase, and we can produce HOCl, which will kill the bacteria. Got it?
Yeah.
Right. Although some of them give us H2O2, some bacteria have the enzyme called catalase. So what this enzyme does is it takes H2O2 and gives us water and oxygen, which is against what we wanted to do, right? We want the H2O2 to create free radicals to kill the bacteria. But if the bacteria can use its enzyme called catalase and nullify H2O2 to give water, which is harmless, it is pointless for us. We cannot create the HOCl, which is needed. So any bacteria that has catalase, we cannot fight if we do not have any NADPH oxidase. Got that? Or is that where...
Understood. Thank you.
Yeah. Shahina, you also?
Yeah.
Yeah. So one of the prime examples for that is Staphylococcus aureus. It has catalase. It will turn H2O2 to water. It'll make it harmless, and hence it has a surviving advantage. And people who have a chronic granulomatous disease will have problems with fighting Staphylococcus aureus. There are multiple bacteria, but I just gave you the most common one. The other clinical correlation that I wanted to discuss was G6PD deficiency. As I've mentioned, G6PD is important for us to regenerate NADPH. And without NADPH, we cannot catalyze the reaction that gives us superoxide. So when you lack G6PD, you do not have NADPH, and you cannot, again, the whole process is obsolete. You cannot do any of what is given there. So basically, all of our cells in a patient with G6PD, all of his cells are vulnerable to free radicals. So it basically cannot handle oxidative stress, because any free radical it encounters, it cannot dismantle it. It cannot fight it. So in a person with G6PD, especially his RBCs, which are prone to oxidative stress, right? Because they carry oxygen, and reactions can take place that convert oxygen to free radical forms, to its free radical forms that can give stress to the RBC. Usually, we have enzymes in place which require NADPH to neutralize the free radicals. But if you do not produce NADPH, you cannot neutralize free radicals, and hence you're more prone to oxidative stress. So in a patient with G6PD deficiency, he lacks NADPH, because of which his RBCs cannot fight oxidative stress, and hence he has anemia. All of the things that increase oxidative stress, like infections and even malaria. So there's anti-malarial drugs which also work in a pathway that increases oxidative stress. It is supposed to harm malaria itself, the stuff that causes malaria, like Plasmodium. But in the process, there's collateral damage. Collateral damage, as in even things that shouldn't be affected will be affected. In a normal human being, who has who has G6, who doesn't have G6PD deficiency, he can make NADPH. He can catalyze the reactions that will help him fight oxidative stress. But in a person with this deficiency, he cannot fight oxidative stress, and hence he'll have anemia and other complications, and he'll be more prone to oxidative stress, which is G6PD deficiency. We will touch on this topic when we teach hematology, but I just want you to know the core concept, which will make it much more easier once it takes place at that point in time. Okay?
Okay, good.
Yeah. Thank you. Yeah, so what free radicals do is just three things that you have to remember. It messes up your lipids, which makes up your membrane, messes up your proteins, and it messes up your DNA. All three things vital for survival, it messes all three of them up. Some examples: carbon tetrachloride is CCl4. It is capable of producing free radicals, and if you don't neutralize it in time, it can be fatal. Hemochromatosis, again, it's a disease in which excess iron is absorbed and stored in our body. One of the places where iron is stored is the liver, and there are multiple other places as well. So when you have excess iron, again, it can react with H2O2, and it can give you a free radical in the form of OH minus. OH minus is one of the most strongest free radicals out there, and it is very highly reactive. Most of the damage that you see, some of the damage that you see in hemochromatosis can be because of the fact that the iron that is in the normal form of Fe2 plus gets oxidized. It loses an electron and it gives it to OH, which becomes OH minus and is highly reactive now. So it can go ahead and damage our cells, and hence damage our cells by causing lipid peroxidation, protein modification, and DNA damage. When we deal with hemochromatosis and see different manifestations of the disease, you will understand, then you will be able to correlate how the iron damages our cells, and hence we get all those manifestations. Like it damages our pancreas and we get diabetes. It can damage our pituitary gland in the head. Lip of your skin. Yeah. And then another concept for which free radical is a co-principle is reperfusion injury. So a lot of tissue, like when a person with myocardial infarction, right, or even atherosclerosis of his leg, when he comes in with decreased blood supply, because of which he's experiencing pain and the skin is getting cold and all the symptoms of atherosclerosis, what physicians do is they try to reopen the vessel and send blood back into those vessels, right? And that's how you get the blood pumping again and get the tissue back to life. But it comes with a disclaimer, like it can be it can damage the tissue further. In fact, there are people. I recently came across an article or something that said more than 50% of the damage that is caused by the injury itself is because of reperfusion when you try to correct the injury. So you have to understand what happens at its core. So when you have ischemia and blood supply decreases, as mentioned earlier, there is problems with ATP production. ATP production happens in the mitochondria. And I'd mentioned that is because of a gradient where there are difference in the amount of electrons and H plus ions in both sides of the membrane. And when these ions move through the membrane, they move the electron through different transporters and finally give it to oxygen. Again, it forms superoxide. So if this reaction is not completed, or if it's not neutralized, or if it's not done properly, you end up with more free radicals. When you do not have enough blood supply and nutrient supply, that's what happens. The the electron transfer chain in the mitochondria does not work well enough, and you end up with more free radicals. And the reactions that is in place for you to neutralize free radicals do not take place because of the lack of ATP itself. And hence you'll end up with more free radicals in that manner. And the second manner is something called nitrogen oxide synthetase, which is present in multiple areas in your body. One of the areas, like for me to explain this concept, you have to know that NOS is present in blood vessels. And what it does is nitric oxide is a vasodilator. So when you have a decrease in blood supply, one of the mechanisms by which our body try to fight it is by dilating, increasing the vessel size. When you increase the size of the vessel, the basic understanding is more blood goes into it. So you have to secrete something called nitric oxide, which dilates the vessel and increases blood supply. But in the process of doing that, even that creates more free radicals, NO minus or nitrous oxide it's called, and which is also highly reactive. So the free radicals that you get from faulty electron transport and from the NO is already present in the tissue that is undergoing ischemia. Now, when you introduce blood into that tissue, this blood is completely full of oxygen. It's red blood rich in oxygen. When you give something as reactive as oxygen to all to the free radicals already present there, it compounds the issue. It adds up, and now you have much more free radicals than before. And this can cause microvascular injury, as in it can cause perforations in the blood vessels. It can increase the gap between each cells in the blood vessels, which will lead to you leaking proteins, leaking RBCs and WBCs, and inflammation ensues, and you can have a disastrous outcome. That is why there is a window within which you have to deal with this ischemia. So take an example of a crush injury, right? Suppose you're part of a deadly accident and your leg gets stuck under a bus, a tire of the bus or something like that. And now you have one of two options, is to wait till a fireman comes, lifts the bus off your leg, and then you tend to the leg, right? Or the second option is you chop your leg off. So what happens is, the more the amount of time you spend under the bus, under the tire, the more ischemia sets in. And the period, as the period of ischemia increases, the chances of your reperfusion injury increases. And after a certain amount of time, even if you reperfuse your blood to that limb, that is your leg that is under the bus, it would just lead to more free radical damage, and it would be counterintuitive. So at that point in time, the wiser thing to do would be amputation. That's what if you, I don't know if you've seen it in any show or come across it in any movie, you will know that people end up losing their limbs in such injuries is because the first responders do not want to risk reperfusion injury. Another example is retinopathy of prematurity. So it's the name of the disease itself is very explanatory. So children usually have to stay in the womb for a certain amount of time. If they do not reach the whole period and they're born before it, they're called premature babies. And premature babies, there's not enough time for your retina to be completely vascularized and have enough blood supply. And if that doesn't happen, and all of a sudden it's ex utero, like once it's out of the womb, out of the mother's body, and in the outside, vascularization does not get completed, and it takes place abnormally. Like once in utero, it's a different condition. There are different enzymes involved, and there's a different temperature, and there's a certain a lot of things have to come together for you to have good vascularization in your retina. But if it is happening ex utero, the vascularization, which is basically building of blood vessels in your retina, is aberrant. It's not normal, because of which the blood vessels are very fragile, and they can easily undergo bleeding and damage. This is one of the main mechanisms by which retinopathy and once your retina bleeds, there can be contractures, and it can lead to problems with your vision. This is the main mechanism by which retinopathy of prematurity takes place. But free radicals again compound this issue. So when you're born ex utero, you have problems with oxygenation, and your lungs are also not that developed. So you supplement oxygen, which can again oxygen at any point in time also can increase like a collateral. It can increase the free radicals. So you have to be wary of retinopathy of prematurity while supplying oxygen to a premature baby. You have to think about the consequences. One is retinopathy of prematurity, and the other one, bronchopulmonary dysplasia, is what happens in the lungs. Same deal, but in this case, because the baby came out too early, it was not mature enough. The lungs was not mature enough. You do not have enough liquid for the lungs to expand and contract as in when we breathe, and because of which you supplement the baby with oxygen. And when you supplement the baby with oxygen, there's always a possibility of new free radicals, which can damage the baby's lungs, and it can cause dysplasia. We had mentioned in the first slide where dysplasia is there's a change in the cell structure which is not adaptive, which doesn't help the baby breathe better. It's just a change because of the stress, and it worsens the problem, right? And it's devastating, both of these conditions. And small anecdote, the reasons why a lot of companies and a lot of pharmacies they promote vitamins, right? Micronutrients like vitamins A, vitamin C, and vitamin E, all of them. The main selling point for all these vitamins in the pharmacy is that they are antioxidants. What they mean by that is exactly this: all the free radicals formed by oxygen, all the oxygen toxicity, vitamins act like catalysts, like the other enzymes that I mentioned, and they help fight this free radical formation by oxygen. Yeah, I think this is Abhi's again. Abhi? What's the first thing that comes to your mind when you think about thyroid? Yeah. Yeah. Do you feel like you can take in more information, or we can do a few questions on what we discussed now, or we can finish this small topic and then do a few questions. We know we are a little over time, but still, it's your call. Do you want to do questions, or do you want to finish a little bit of micro as well? We'll keep this quick, I guess. That is if you want it.
We can do the questions.
Do you want a break now, or do you want to jump
Yeah,
into
after the break would be nice.
Okay. 5 minutes?
Yeah, okay.
Oh shit.
Can we start?
Yeah.
Yeah. Can we try ourselves first?
If you have anything like, can you, if you could just type it out, it's
Is
we'll
it
know.
E pleomorphism?
Okay. Anybody else?
I think so. E.
E again. Okay. Others, do you want to take a guess, or should we just move on to the explanation?
Only one? Maybe the dysplasia? No.
Jairo, what's your answer? Could no, he has he has no, his kidney is like there's a blockage in the kidney. Like yeah, he's not getting blood supply to the kidney. Ischemia in the kidney.
necrosis, bro.
So guys, do you have an answer? So yeah, Sahina already mentioned. She said it was B. And anyone else have any ideas?
Not sure.
Okay, so yeah, let's go through this question. So, 65-year-old man comes to the emergency department due to acute onset severe right flank pain, nausea, and vomiting for the past hour. Uh, his medical conditions include prostate cancer and membranous nephropathy. Okay, he already has some problem in the kidney, and he had prostate cancer before. So, so just from these symptoms, we know that this probably might be related to the kidney. Might not, we cannot say for sure, but since he has right flank pain, so his kidneys might be involved because kidneys are usually in the right flank area. Um, so then moving on, blood pressure is 148 over 60 and pulse is 95. It's normal for a person with with pain, so vitals look normal. Then on physical examination, the patient appears to be in moderate distress due to pain and is diaphoretic. So while looking at the patient, you know that he's very uncomfortable because of some pain, and he's sweating a lot. That's what diaphoretic means. There's a right costovertebral angle tenderness. So right CVA tenderness. So I'm sure you guys might have learned in your propedeutics or in your physical examination classes that to check for anything wrong with the kidney, maybe some inflammation or some acute process in the kidney, you do the CVA tenderness. Basically, if this is your right side over here, you would hold it and you would hit it from the back just to see if there's any pain or something. So he does actually have pain over his right CVA. So definitely now we can think of kidney. And then they did a urine analysis and it shows blood in the urine, hematuria. So we need to know what's wrong with the kidney, and that's what they have given us after that. They did a contrast enhanced CT scan, reveals a wedge shaped perfusion defect in the right kidney. So this gives us a lot of information, just this one sentence, that the CT scan reveals a wedge shaped perfusion defect, meaning there's no blood supply to one part of the kidney. So if you remember in one of the previous slides, I showed you a picture of like a piece of a kidney with had some type of necrosis, and it had a perfusion, like I told you, some artery was probably blocked. So that type of perfusion defects usually result in a wedge shape because the blood vessels spread throughout the kidney. They start at one point and branch off. So if the blockage is in the beginning, it's more likely to produce a wedge shaped defect. So just from that, we know it's some kind of ischemia is happening. So that's what we have so far, some ischemia in the kidney. The affected renal tissue is most likely to develop which of the following histological changes over the next several days? So just for answer choices, it's all different types of necrosis. So we'll see. We'll just look at what we have so far. We had ischemia of the right kidney. That's what we think because of the wedge-shaped perfusion defect, the pain, hematuria, and all that. So there's some blood loss to the kidney, and the ischemia will eventually result in coagulative necrosis that we saw before. So all organs, when they undergo ischemia or infarction, they will undergo coagulative necrosis with the exception of one specific organ. Do you guys have any idea what that organ is? I had mentioned the exception before when we were doing coagulative necrosis.
Brain?
Yeah, it's the brain. So as in the brain, there it actually undergoes liquefactive necrosis because remember I told you guys about the neutrophils coming in and then liquefying the coagulated proteins and digesting the material. So that's so again, if you see ischemia in any tissue except the brain, you should be thinking about coagulative necrosis. So that's all. Like all this fancy big presentation, and all that was necessary for you guys would be to just read this line, which shows CT scan reveals a wedge-shaped perfusion defect in the right kidney, meaning there's loss of blood supply, so there will be ischemia. Then they want to know the histologic change. So you know ischemia in any organ except the brain results in coagulative necrosis. So this question can basically be cut down to just these two lines. But yeah, they like to confuse and distract a lot. The answer choices that UWorld has, yeah, they just explain the different types of necrosis. They have a nice table about this. So liquefactive necrosis, again, we saw in some types of bacterial abscesses or infections, and or wet gangrene, and CNS infarction. So when CNS loses its blood supply, you'll have liquefactive, and hydrolytic enzymes digest all that stuff. Fibrinoid necrosis, again, blood vessel related. We saw it was malignant hypertension and vasculitis, uh, fibrin deposition in the vessel walls. So pretty similar. Fat necrosis, we saw trauma to the breasts. Breast tissue can cause trauma, and then we also saw a picture of that obese patient with fat necrosis with a lot of red bumps in the back because of subcutaneous adipose tissue. So it can also cause fat necrosis. Again, the same mechanism. They have mentioned free fatty acids bind with the calcium, and they form those white chalky things that even on X-ray will be visible. Caseous necrosis, again, fungal infections, mycobacterial infections, and nocardia, I think. Yeah. These, no, sorry, not nocardia, but yeah, these fungal infections, mycobacterial, tuberculosis infections. I told you, yeah, they look like that white cheesy material because of all that lipid and fat that these guys need to survive. Um, in addition to the dead cells as well. Then coagulative necrosis, which is irreversible ischemic injury to any organ outside the CNS. So that's what basically they were looking at. Again, coagulation, the same process. The idea is loss of blood supply. You cannot make ATP. No ATP. You cannot produce. You cannot yeah, you cannot undergo aerobic respiration. Then if aerobic respiration cannot happen, you switch to anaerobic. Then lactic acid gets built up within the cell. So acid causes denaturation of all the proteins in the cell. Then they start clumping together. Then they, yeah, basically that's it. That's, and then they coagulate. Clumping and coagulation. So that is coagulative necrosis. They maintain the architecture, but the denaturation is what is the main part. And then eventually there will be infiltration over time with uh, neutrophils and macrophages. Again, that's an important difference between necrosis and apoptosis. Necrosis is followed by acute inflammation because it needs a cleanup, versus apoptosis is programmed cell death. It's like suicide, so need to clean it up, I guess. Yeah. um yeah, those are the main points. Again, same things they mentioned over. Yeah, the other answer choices. So Charaka had said A, caseous necrosis. So yeah, it's usually found in mycobacterial or some fungal infections. And I think fibrinoid necrosis was also mentioned. It's a histologic pattern of injury. Yeah, fibrinoid necrosis. We saw that it mostly, yeah, okay, because ischemia, you could have correctly thought of fibrinoid necrosis, but the thing is, it if it was vasculitis, meaning the cause of fibrinoid necrosis, it usually affects multiple organ systems, and here they clearly mentioned that it was just one part of the kidney, so it's more likely to be some ischemic, some kind of emboli or something like that blocking that led to this condition. So fibrinoid necrosis, yeah, it's not dystrophic calcification, and fat necrosis, no. All right. So do you guys want to go for one more question?
Maybe just one more. Is that okay, guys?
Yeah, sure.
Yeah.
All right. Ashwin, do you want to take this?
I'll do one. Yeah. Dude, can you stop presenting then?
Yeah.
Right. So this was a topic we discussed, right? I'll give you some time to go over this question, and we can discuss the answer.
Can you zoom in?
You guys ready? Okay, so we have a couple of people saying D, right? So we'll go through the question. A 74-year-old woman comes to the emergency department. For 2 days, she's been having left leg pain and numbness. She has a history of atrial fibrillation and hypertension. So atrial fibrillation is a type of arrhythmia. Arrhythmia is when heart is not has a disorder of beating not in rhythm, right? It's not beating in rhythm, so there's an electrical issue. Her left lower limb is pale and cold to palpation below the knee. Ultrasound shows an occluding thrombus, and a left femoral embolectomy is performed. Post-procedure, dorsal pedis and posterior tibial pulses become palpable. Several hours later, the patient begins to experience severe left calf pain. On repeat examination, the left calf is swollen, tense, and tender, and the pain worsens with dorsiflexion of the left foot. The distal pulses are also absent, and the physician suspects injury of the reperfused muscles. Wait a second. Reperfused muscles in part due to overproduction of reactive oxygen species. Which of the following enzymes is most likely to help neutralize these toxic molecules? So, just like the previous questions, you could get the answer from the last two lines, but I'll explain the clinical vignette first and we can come to this. So what happened is this patient has atrial fibrillation and hypertension. In both of these, so hypertension is one of the causes of atrial fibrillation. And in atrial fibrillation, as you know, the heart has 4 chambers, 2 atria and 2 ventricles. In atrial fibrillation, the atria is not contracting properly and it is not in rhythm, because of which there's an increased chance of the blood to blood stasis, right? For the blood to get caught up in the same area. And when that happens, there's more chances of you forming a clot. When you form a clot and the heart pumps the clot to different parts of your body, if it's in the left side, it can go on to travel to any part of your body. And in this patient, the clot traveled and it kept traveling down to your leg, to her leg, and it crossed a thrombus, which means there was an artery in your leg. The clot traveled all the way from the heart, reached your leg, and blocked that artery. So now anything beyond that artery is not getting any blood. So the procedure that the doctor performed is called an embolectomy, where they went in and broke the clot apart so that they can revascularize the vessel and now blood can flow through it. So after the procedure, when they so palpation is when you keep your hands on the blood vessel and try to see if you can feel the artery move up and down, right? So when they did that, they could palpate a good pulse beyond the place where the clot was there. That means they were successful in their surgery and now the blood was going back to that limb. Even though that happened, a few hours later, the patient is experiencing extreme calf pain, right? And now it is swollen, as in it's become bigger. It's become really tense and tender. Tender as in it's extremely painful. And the pain increases when the patient is dorsiflexing. Dorsiflexing means if suppose this is your leg and this is your ankle, this movement is dorsiflexion, right? You're moving it towards your body. So when he dorsiflexes, the pain worsens. This is one of the manifestations of reperfusion injury. And the doctor is also suspecting reperfusion injury. So as I mentioned before, reperfusion injury is caused by formation and overproduction of free radicals. In other words, reactive oxygen species, right? It's just another way of saying that. So they're asking which one of the following enzymes will help neutralize. So this is one of the scavenging enzymes that I had mentioned that works against, that tries to neutralize these molecules and saves us, right? And let me just see what the answers were. So we have a couple of people saying D and one person saying E, right? So even if you don't remember completely the reaction of what takes place, do you recall that? Okay, let's go through all the options, right? So myeloperoxidase is one enzyme that maybe you wouldn't know all the enzymes in the answer options. So we'll work with what you know and what was introduced in this PPT, and then we'll look into the other options. Myeloperoxidase. If you recollect, this was the last step in the process that led to the formation of HOCl, which helped kill the bacteria. So if it helped kill the bacteria, it means it was a free radical, right? So that enzyme was responsible for creating the free radicals. So obviously it didn't help us neutralize anything. In fact, it would have made it worse. So myeloperoxidase cannot be the answer. NADPH oxidase was the first enzyme that changed oxygen to superoxide, which is a free radical. Remember? So if that was causing free radicals, there was no way it'll help us neutralize toxic molecules. Let me see if I can. Oh, wait a second. It'll be better if I stop presenting. Go back all the way. Right. Check this out. So NADPH oxidase catalyzes the first reaction which converts oxygen to O2 minus, which is a free radical. So the question was, what helps us neutralize these toxins? So by this logic, NADPH oxidase can also not help us neutralize this toxin. On the other hand, option number, the correct option, which was superoxide dismutase, is what it does is it catalyzes a reaction that turns this superoxide to H2O2, which is not highly reactive, which is not a toxin, which is not harmful. So the superoxide dismutase is one of those scavenging enzymes that helps us neutralize the toxin, which was exactly what the question was looking for. Okay, was it this? Right. So you had only 3 enzymes to work with because we had not introduced the other 2. But if you had recollected that myeloperoxidase and NADPH oxidase were responsible for making the free radicals rather than neutralizing, you'd have only been left with superoxide dismutase, which is the answer. So this syndrome, what this patient is experiencing, is called compartment syndrome, is because the tissue swells up and there's not enough space. So there's so much pressure from the muscles around the vessel that the muscles contract and the vessels compress, because of which the blood supply is broken to that tissue. And the pressure keeps on increasing. That's why this patient was complaining that his tissue was tender, tense, and swollen. Because of the increase in the size, the blood vessels get compressed, and the blood supply to that organ decreases. In fact, the way you treat these patients is by making a cut on their skin so that you let the pressure out and the blood flows outside their body. So you didn't have to know that to get the answer. All you had to know that was that when you introduce oxygen into tissue that was ischemic, there are multiple mechanisms, one of which is NADPH oxidase and the other one nitric oxide, which we had mentioned previously, that are responsible for creating free radicals like superoxide, hydroxyl, and oxygen. Single oxygen. So all of this is responsible for further damage. So we need enzymes to neutralize this reaction, right? Out of which the only option, I mean, there are 3 enzymes that I mentioned here: catalase, glutathione peroxidase, and superoxide dismutase. But the only one in the options was superoxide dismutase, right? So that was the correct answer. That would have converted our superoxide, which is O with 2 minus on top, to H2O2, which is far less reactive. They just go on to say that, right? To state what we have already discussed is the fact that reactive oxygen species are continuously generated every time the reaction takes place. But we usually have antioxidant enzymes like the ones mentioned that help us fight the oxygen species. But in cases where we have excess of these free radicals and reactive oxygen species, we are outnumbered, and the antioxidant enzymes are not enough in number, and that's when you can cause cell injury and cell death. So that's how you should have landed up to the answer. Now for the 2 enzymes that we have not discussed, which was part of the answer choices. Cytochrome oxidase, we touched upon the electron transport chain in the mitochondrial membrane that is responsible for making ATP. Cytochrome oxidase is one of the last components of that chain, which functions to create the gradient and transport the electron and end up in making ATP. So basically, it is no way involved with either being creating or fighting ROS. Cytochrome P450 is once we learn about the liver, you'll come across it. what it does is it metabolizes a lot of toxins that we ingest. Even the drugs that we ingest can have reactive forms. Like if chemical reactions take place and some of the bonds are replaced, it can become very reactive and harmful. So cytochrome P50 is a group of enzymes that catalyzes reactions to metabolize toxins and drugs. That's all you had to know. Myeloperoxidase, as we mentioned, creates HOCl, which is hypochlorous acid, which is more reactive and it can cause damage. The same goes for NADPH oxidase. It creates superoxide, which is also reactive.
Excuse me, can you zoom your slide? It's so small.
Sorry. How about now?
Thank you. Yeah.
Were you guys able to follow me?
Yes.
Yeah. Thank you. So I think some of you did get the answer right, and it was superoxide dismutase. We had a couple more questions, but I think we are already past our time, so we can wind up for now. Abhi?
Well, I would say you guys did a pretty good job with the lecture, even though I lost my attention span. So, mm, um, so the thing is, what suggestions do you have for students who are like below average? Because I would say currently I'm a below average student with the knowledge I have.
You, so the thing is, you still have time, right? One of the good part about the step one exam is that you can schedule your own exam. They don't have fixed dates, so you can take it when you feel you're ready. So if you're starting now and you feel you need extra time, you can delay an exam by a bit. But we are starting from scratch, so even I wouldn't say like if you're completely new, you would still you completely use it'll be informative for you as well. But yeah, if you're going to go parallelly with us, then in the span of 6 months, you should be ready. So we're not targeting like the students who are extremely like highly achieving or something like that. This lecture is for anybody. We'll cover every portion and we'll do it as many times as you want to make you understand. And once it's done, you should have a pretty solid base. And from there, you can take your time yourself and schedule your exam when you feel ready.
So, how was your plan like for the classes again? Like, is it like depending on specific student or like as a whole?
You mean timings and stuff like that, or?
Yes, yes. Is it like flexible or like in a particular time?
No, it's flexible. Like, see, right now I see 4 people who are part of this other than us. So it's a small group, right? We can come to a consensus on like based on your schedule and ours, which is comfortable, and we can work with it. It's not very rigid. It's just us. So yeah.
Okay.
Others, anything that you'd suggest, any way you think we can change to make this better? Questions more, or diagrams, or explanations? Or...
This way is pretty good, I feel.
If you do find some informative videos, you could play them also, just to get a good view.
Yeah. So, did you mean you wanted the video to be part of the class, or you just wanted access to them?
No, part of the class. Like, in case there are some really good YouTube videos like that can help with more visualization aspect of learning. So you could include that if you knew any good videos.
Yeah. So we were thinking initially that if you had a background, like if we freehand give you what to study for the class, and you had some sort of a background, then we can use this time just to brush the concepts and do a lot more of questions and stuff like that. But if you need the videos to be part of the lecture, then sure, then we can do. Anything else, guys?
I feel like I need to exposure to more questions because I'm not familiar with the question. I can able to understand theories, but question is the most important. So please add some more questions and please discuss as much as possible.
Abhi, we can't hear you.
And another thing about the timing as well. Sometimes if the time exceeds, it can be a problem for us, like to study for the other things that we need to do.
So can we get these PPTs for preparations somehow?
Yeah, so we're thinking about it. If you like, we'll let you know maybe by today evening or tomorrow morning.
Yeah.
Is that okay?
Yeah, of course, no problem.
Yeah. So the thing is, guys, so there's a certain amount of time that goes into preparing the lecture and the questions and putting up a presentation together. So to make all of this effort worth it, we at least need a few more of you to be a part of this. Only then can we have a wide enough range, and make we'll feel it's better use of our time if at least maybe 10 of you are part of this. Right now we have 4. If so, if you feel others will be interested, anybody who you know, and if you feel that you like the lecture now, can you spread the word with your friends who are planning to take the step anytime
Sure,
soon?
sure.
Yeah, thank you. So we'll end up doing the same amount of hours of class. It's just that we'll be doing it in 6 months, and they'll be doing it in yeah. There was yeah. So about the next class, so the 4 of you have joined, and if anybody else does join, maybe we can put up a so regarding the day and date in which you want to do the date and time in which you want to do the next class. Probably put up a poll and find out a convenient time for all of us, and yeah, majority wins.
And
That
how
okay?
can we do that? Like,
No,
right
I'll
now?
put up a poll, like just how the registration worked. So I'll put up a poll asking which day of the weekday that you would prefer, and we can come up with a date that most of you prefer, and we can do it that way.
We will be sending it to everybody's email, yeah?
Yeah. We'll send it.
I think my email ID is not added. Could you please add this email too in the
Yeah,
list?
sure, done. Yeah. Anything else, guys?
Thank you, guys.
Thank you for your good job. Thank you.
Thank you.
Man. Yeah, what do you think? Dude, I can't hear you. I can't hear. Yeah. No, no. I thought Charaka was Rashad. No, Charaka was Rashad was like, I am in there with somebody else's account. I thought that was Charaka's. Oh, okay, okay. Dude, did you like it? Yeah. I saw that. Yeah. Thank you. Not looking. So yeah, I was thinking of that during the class. Dude, did I say something wrong when I was stuttering in the beginning? Like, did I give wrong information at all? Yeah. I slapped it. Yeah, but other than that, I didn't tell anything wrong, right? Second time was much better than the first time. Like when I started talking, I think I was slower. Yeah. Dude, why did they leave, man? Do you want? Nobody was answering, yeah. But I think all of them are doing it like for the first time ever. These guys, yeah. Oh, that can work. There's also, yeah, for sure. Yeah. Uh, okay, so we need at least 10 to go ahead with this, right? Three won't be ideal, right? Malika will join, right? Yeah, those were like how